Danny, can you hear me?
I can hear you.
Oh, hi. It's great. Over the music. How are you guys?
Doing well. Good to see you, bud. Thank you.
Same here. Thanks for coming. Hi, Peter. How are you?
Well, this is great. So when we talk, the music actually goes down. So maybe we just need to keep talking.
I know. Hopefully, I think it'll be pretty seamless. It's amazing the things that I notice with Zoom. If you share a screen and then you put something else on in front of your screen, it'll only share that. It won't share the Zoom controls or anything, which is, I found that out teaching the other week. So
I don't know, this post-outbreak world is all kind of...
Yeah.
Laura, how are you? Where are you? Are you not have your camera on? Oh,
Wait, where's Dolores?
Live on Facebook.
I can't see Dolores. Where is she?
I don't know, she's hiding.
Oh, there she comes.
Oh, really? Oncosome pictures, Dolores? Seriously?
Hi.
Hi, Dolores. How are you?
Good, nice to see you. Thank you for organizing this.
No, I really appreciate all of you guys coming on. I think it'll be great, and you guys are awesome. So, oh, there's Alana.
I just wish I could be
Hi,
on all day.
Alana.
How's it going?
Hi. Good.
Hello.
I don't know.
Hello. Good to see you all.
Same here. Okay, we have everybody in the morning panel is here. Awesome. So I'm gonna.
All has happened.
I know.
Surprised. Josh, you sound surprised. Come on.
No. Because the whole, the East Coast, Central Time and everything. I just wanted to make sure everybody was, was on. Pat is, I think, here earlier than, or did she leave? I think, no, she's still here, right? But it's okay. Pat's going to probably be here the whole day.
Hello,
maybe.
I'm just listening in.
Okay, Pat, you're not on for another 2.5 hours, just to let you know.
I know, but I wanted to see what everybody else is saying.
Okay, good. No, good. I just wanted to make sure. No, we love having you. Don't worry.
Do we need to do sound checks or video share screen checks now, or?
Yeah, so let's, can you just all make sure you have access to share screen? Why don't we go ahead and, we have 5 minutes. Danny, do you want to try to share your screen? And then we'll just go through. Awesome. Okay. And now why don't you stop? There we go. And then Alana, why don't you go next?
Okay. Make sure that works.
Awesome.
Are you seeing the full screen or my presentation mode?
Now I see your full screen.
Okay, good. Because sometimes, sometimes when you have two monitors, it picks the wrong one.
Okay, Peter, go ahead.
You're stressing me out.
Stressing out the physicist. There we go.
I know. Technology. I don't do this technology thing. There we go.
There we go. Good. And this is being broadcasted, so right now, so it's okay. Good. Hi everybody. We're doing our pre-check to make sure everything is going well for everybody, so that we can give you a great morning conference. And Dolores, why don't you go ahead? Perfect.
At this point, presentation.
Perfect. Okay, good. We're doing it. And I already tested mine out earlier.
Hey Josh, do you have a link for the Facebook live stream?
I don't. Darren, can you put the link on our chat room if you can for the Facebook Live? So Darren is going to he's our master ceremony technologist in the back room. He'll put it hopefully into the chat room.
Oh, I think I got it.
You have it?
I just saw my own slide. That's weird on Facebook Live. Oh.
He just, no, oh, that's.
Josh, how many participants are there?
So on the, well, we have 8 here, but then this is going through Facebook Live, and I think a lot of people are viewing it through that, and I don't know until we log off or I can see the Facebook Live, but it got, yeah, so hopefully we're gonna have a big audience. It just depends, you know, there's 3 or 4 different ways people can log in, right? So what you see on here is not necessarily the right number.
Mm-hmm. Mm-hmm.
Because this gets fed to everybody. So just some housekeeping. At the end of this, we're gonna the 5 of us, and I think Danny has to go to another meeting, but the 4 of us are gonna do a panel discussion like I mentioned, and then I'll moderate we see the questions on the chat, but I'll moderate that. So for you all. Yeah, this is just, I mean, the positives of COVID are that I think we are now going to experience a whole new way of conferencing, which is great.
Mm-hmm.
I miss all of you guys in person, but
yeah, me too.
Likewise.
It opens up to, you know, a world of
The world.
Yeah, it does. No, literally, I was gonna say it's a world of, but I'm like, no, it is a world.
We're doing a cancer biology training thing. That's the other meeting, and I'm the treasurer, so we're enjoying it, but at the same time, you just don't get those interactions with other people.
Yeah.
Because we're not as technologically advanced as Peter.
You guys all see my screen, right? Good.
Mm-hmm.
Okay, I think we are on it, right? In 16 seconds, we're gonna be on. Okay, are we on? Darren, are we on? Because the countdown is at 0.
Yeah, you're on.
Okay, perfect. Good morning, everybody. I hope you are all or good afternoon, depending on what part of the country or actually the world you're at, right? So it's morning here on the West Coast. My name is Josh Neiman. I'm an assistant professor at USC, and I'm the moderator for today for our Metastatic Breast Cancer Research Day. I want to thank, if you were on earlier at 9 o'clock or, about an hour ago, I want to thank Sharon and Linda Tantawi, who have been doing a fantastic job of hosting this 1-month conference from LA to New York, Metastatic Breast Conference. Today, I'm really excited and delighted to have my colleagues joining me for this conference. And the goal of today's conference is basically, it's for you. It's to educate the metastatic breast cancer community on the latest research, and breakthroughs that we are having, and educate you so that you can go away from today feeling a little bit more knowledge about whether it's your disease or a family member's disease. Just a little bit of an overview of how today will happen. Today is broken down into 2 sessions. Basically, the researchers that you see, and I'll go through that, introduce them, will be in session 1, and we'll have 2 hours in the beginning. And at the end of Dr. Davisio's talk, we will have our panel discussion with the 4 of us talking and answering your questions, which I will moderate through the chat room. So if you're on Facebook Live or on Whova or on this webinar link, go ahead and post your chat, and that is going to get fed to me, and I'll bring it up. The topic today that the theme of today is really building up from the basics of metastasis all the way to clinical trials and cognitive issues. And I'd like to go through the first session, and just introduce speakers, just very lightly. I'll give a real introduction after or before they're each speaking. But our first panel, we have, including myself, we have Dr. Danny Welch from Kansas, who's going to be talking about the hallmarks of metastasis. And then following will be Dr. Lana Well from Huntsman Cancer Institute, talking about her work in functional precision medicine, and follow up with Dr. Peter Kuhn here from USC, who's going to be talking about his fascinating with liquid biopsies. And last but not least for this morning, we'll have Dr. Dolores Davisio from Cedars-Sinai talking about extracellular vesicles, which are those packets of information that cells send out. One minor change to our program this morning is that Dr. Danny Welch is going to be going before me. And that's because in the world of Zoom, he has his own conference that he has to go to. But so to accommodate him and really, we wanted him to, to be at this conference, and see as such a perennial researcher. And I know Danny hates getting, being put on a pedestal, but he is. But we really wanted him to be here this morning. So just one minor little switch we had this morning, but everything else hopefully should be seamless. So with that, I want to get to the crux of everything. I'm going to introduce Danny. Let me stop sharing my screen. Danny, why don't you start putting up your screen before I introduce you? So Dr. Danny Welch, many of you in the metastatic breast cancer community and in the metastasis field knows him. He doesn't need an intro, but I will give him the intro that, for those who don't. Dr. Welch is a founding director of the University of Kansas's Medical Center's Department of Cancer Biology. He's also a professor there in the medical school, and he's a director of the NFCR Center for Metastasis Research. His research is in the area of metastasis suppressor genes and the biology of metastasis, and he is best known for his discovery of the 4 of the 24 well-known metastasis suppressor genes. These are basically genes that stop the growth of the metastatic cells, and I'm sure he'll talk about it more. He's a previous Komen Scholar, and he has authored over 140 original research papers in the area of cancer biology and metastasis. And his research is focused again on unraveling the biology of metastasis. And Dr. Welch is an amazing mentor. And I have to tell you, I'm one of those folks that he has mentored along his career, and I'm very thankful for him to be here. And without further ado, Danny, please go ahead.
Well, Josh, thank you. And thank you for being so accommodating with trying to juggle multiple schedules. I am honored to be leading off this morning a group of speakers that you're going to hear a lot of details on. And I'm taking my role as the person who will sort of set the foundation upon which you'll see a bunch of details and exciting research that each is doing. And with that, I'll just sort of help get started. I'm teaching medical students and graduate students, and one of the first things we have to explain is why cancer is so stinking hard to cure. And it's because the cancer cells, the drugs that we're using are not entirely specific to the cancer cells. We're trying to develop therapies that are more specific and don't give all those off-target side effects that are so debilitating. In addition, the cancer cells are not the same. Within a tumor, there are thousands of different subpopulations. Some are resistant, some are sensitive, some are able to spread, some are not. But ultimately, the phenotype or the characteristic of cancer cells that's most responsible for cancer-related deaths is metastasis. And so a couple of little facts, it's that 90% of the deaths or the bad side effects directly caused by the cancer are associated with metastasis. The good news is in 2020, despite it being a weird year, it's not an automatic death sentence, which is unfortunately how it was talked about back as little as 30, 40 years ago. We do not yet have a cure, but there are significant advances, and it's not uncommon now to meet people who are living 10, 20 years post-diagnosis with metastatic disease. Unfortunately, despite the magnitude of the problem, funding has not kept pace with the issue of what metastatic disease is. So the key take home that I'd like to go away with before I start showing the process and the hallmarks is that metastasis and making a cancer are not the same thing. They are related, but they're not the same. And it's not realistic to treat cancer in the same way for the primary tumor or the metastases. And in a way, you can look at it this way. Bob Weinberg and Doug Hanahan defined what they call the hallmarks of cancer. And you can look at that as this big black ball on the side. Doug Hurst and I tried to look at the hallmarks, those characteristics that are present in every metastatic cell. And those are the ability to move or invade, to screw up the microenvironment wherever the tumor cells are, and the ability to adapt and then grow somewhere else. And so those form a subset of cancers, and a subset of the metastatic cells are those hallmarks that determine where the cancer cells will grow and populate. So how do you get these metastases? The cells can spread through any sort of highway system present in the body, through the lymphatics, through the blood, or even across body cavities. In the case of breast cancer, it's the lymphatics and blood or hematogenous metastases that are primarily responsible. And I've generated a series of animations, and I'm going to go through this rather quickly because you have other people who get into the details. But hopefully you'll see part of the reason metastasis has been so difficult to treat and understand. It begins with the growth of the tumor. That's pretty obvious. And the tumor cells are mutate such that a subset of them gain the ability to do what normal cells don't do. In the case of breast cancer, breast cells don't move around the body. But those cells in the breast that have become tumor begin to communicate with other parts of the body, such as the bone marrow. And they do this via these little sacs that contain DNA, RNA, protein that actually mobilize the stem cells that generate the blood cells in our body and many of other tissue types. And those stem cells move around the body, get to other places, and they create what's known as a pre-metastatic niche, kind of like a vacation home for a cancer cell. So they're leaving the urban breast and going to a vacation home in the brain, liver, lung, or other organs. And a lot of studies are taking place to characterize these exosomes and the communication that's taking place. After the cells communicate, they begin to walk or migrate. Just like these dogs and this little baby, they actually have to learn how to crawl. And the difference is that you can crawl by different mechanisms. 20 years ago, we actually used to just think of crawling or migrating as one thing. But over the last couple of decades, the ways in which cells move have been better categorized. Some move as a collective, as you see in the top. They move as a group of cells. I kind of liken this to teenagers at the local mall when we could actually still go to the mall. They move as a pack, disrupting everything in their path. Others move independently. That's in the bottom left corner, mesenchymal. This is a sort of that loner cell, but it's got hyperactive abilities to move. Others are a little bit more subtle. They independently move, but they do so without being noticed so much. They're amoeboid. They're kind of like amoeba. They shapeshift and squeeze through the collagen that makes up the tissue. And still, the fourth mechanism, originally described in the late 1950s for certain white blood cells, but for cancer cells, it's been called entosis. And this is when a mobile cell goes through another cell and appears on the other side. So each of these involves literally tens to hundreds of different molecules that have to be expressed in a coordinated way. Then, as they're doing this, people have been showing that the collagen, here you can see it in fluorescent green, gets reorganized. And you can see it's kind of like a chain link fence here. That's normal. But after the first cells have gone through, the collagen gets realigned and it's put in parallel. And you can see electron micrographs showing this. And what that basically leads to is early cells move very slowly, like pioneers moving from the East Coast to the West Coast. And unfortunately, Josh, this is only at a 2 a.m. LA freeway kind of view, but the first cells that move only a couple of microns or 1/1,000,000th of a meter a day. But later, after the collagen gets reorganized, they're moving that same distance in an hour. So it's much more sped up. And the cancer cells, again, are manipulating their environment, both by communicating to the bone, but by reorganizing the matrix. Recently, the last couple of years, it's been shown that the tumor cells are recruiting different types of neurons or nerve cells, and those are causing changes in the behavior of the tumor cell. The actual mechanisms responsible for how the neurons are regulating the cancer cells, I don't think are particularly well characterized, but this plays very well into the kinds of studies that Josh is doing and Pat Stieg are doing with regard to brain metastases. Tumor cells also recruit in inflammatory or immune cells. Those immune cells squeeze between the lining of the vessels. They begin to populate the various parts of the tumor, and those inflammatory cells can attack the tumor, but also promote the behavior of the tumor. And the ones on the left are macrophages, a type of cell that eats others. And on the right are what's known as neutrophils. These have very weird looking nuclei. And one of the speakers this afternoon, Cyrus Kajar, shared this video with me, but it shows how quickly the neutrophils can attack something that they recognize. In as little as an hour, literally, you have all those neutrophils coming from the blood and attacking. Eventually, those immune cells and the tumor cells form a conga line. And although he's not speaking here, John Condelis and his group have shown that it's an alternating tumor cell, immune cell, tumor cell, immune cell. Think it sounds like a conga line, tumor cell, immune cell. And the immune cells, in fact, are doing a lot of invading for the cancer cell to allow them to move, in this case, into a blood vessel, but can also move into a lymphatic. And as they're doing this, remember the cancers are growing behind them and they're actually pushing the tumor cells towards the bloodstream. And it's a little like trying to get into a Japanese subway. The cells behind are actually doing some of the pushing to get more and more tumor cells into the bloodstream. And the number that will actually enter the vessels or intravasate can be somewhere between 1 million and 4 million cells for every pencil eraser sized tumor every day. And that's really kind of a depressing statistic. But the good news, if we can take the silver lining, is that we don't see millions of metastases. We really only see very few. It's a very inefficient process. And what's been recognized, and you'll hear more from this from Dr. Kuhn, is the cells, while they're in the bloodstream, can be characterized. One can look at the behaviors of those cancer cells and also the DNA that's in them and released from them that maybe provides information that will help physicians diagnose early, detect early, and treat more appropriately. And here's an example of some of that kind of characterization. It's just one of thousands of different ways people are looking at it. But you can have cells in breast, they're normally epithelial in origin. And there's another type of cell that occurs in an embryo, and they're known as mesenchymal cells. And there's a way during the development of an embryo where these can convert back and forth. And cancer cells can have epithelial, mesenchymal, and then mixed properties. And even as little as 5 years ago, people thought there was an epithelial cell and there's a mesenchymal cell, and they were distinct. Now we know there's at least 100 different variations, 1%, 99, 50-50, and so on. So the complexity just continues to grow. Those cells that are in the bloodstream can begin to make friends. They sort of accumulate a posse. They'll cluster with each other, homotypic, or they can cluster with other things like platelets or white blood cells, or even the material that forms a clot known as fibrin. While they're doing this, they're evading host immunity. They're just escaping the immune cells that are trying to attack them. And they're also changing their properties to hide, like Harry Potter and his invisibility cloak, finding ways in which to just completely hide themselves from the immune system and the shear forces that exist in a blood vessel. Remember, the cells are going, but the liquid that's part of the plasma that goes by them is actually pulling against them, much like the air is pulling on these dogs. And that shear force varies in different parts of the body. And it turns out, for the most part, the cancer cells, when they adhere at various different places, tend to do so on the venous side because it's a lot less shear. And as they get to a different place, they'll adhere, and then they start to squeeze through the very small spaces between endothelial cells to escape, get out into the tissue, a process known as extravasation. And that's where they encounter that pre-metastatic niche that started a whole lot of this. And some of the pre-metastatic niche are some of those neutrophils that I had mentioned before that eject some of their DNA that somehow the tumor cells take up and recognize, change their behavior, and it allows them to begin to grow. And that can even precede development of new vessels by many weeks or months, so that they basically take over already existing cancer or blood vessels to supply them with nutrients. So that's sort of the process in a nutshell, but I want to just highlight a couple of individual points, because this afternoon you're going to meet the lady who actually started the field of metastasis genetics, and that's Patricia Stieg, or Pat. While she, like I, have been studying mostly metastasis suppressors, there are genes that promote the process. And you'll hear some of that from a couple of the other speakers. But these are things like the accelerator pedal in your car. They're the ones that drive the cells. Suppressors block the ability of cancers to metastasize, and they somehow get turned off in a metastatic lesion. And a new category that's emergingly recognized are known as the Metastasis Modifiers. These are like the transmission in the clutch. You can have the gas, but in first gear, you can only go so fast. In fifth or eighth gear, you can go faster. And so these are modifiers of both the suppressors and the promoters. And various groups are studying those. We're very interested right now in the modifiers that exist in different races. And this might explain why cancer is more aggressive in certain races of people versus others from a genetic level, notwithstanding all of the socioeconomic issues that exist. And finally, I think all of us, I know everybody who's speaking is 100% committed to the priorities of the metastatic patient. Our goal is to give you a cure and highest possible quality of life. And these were the 5 areas that were identified by surveys and conferences as the top 5 areas that metastatic patients wanted to see addressed. And you're going to hear actually about all of them at some level during the presentations today. We want to make sure that there's complete access to care to give the highest quality and quantity of life. Detect it early, treat it more effectively, and wherever it occurs, and give people the best palliative care possible. And with that, the last thing is, here is what it looks like for cancer cells to spread. One of our goals is to freeze those cells so they can't leave in the first place, or they get halted wherever they are. So Josh, I think I've got a couple of minutes for questions if we have time. Thanks again to everybody for listening, and thank you for the invitation.
Thank you so much, Danny. Yeah, so since you won't be part of our panel, we have a couple of minutes to, if anybody wants to ask Dr. Welch any questions, please put it into your chat box. We'll have at the end, we'll have a moderate, all of us coming together and answering questions at that point. But because Dr. Welch has to leave, we have a couple minutes. We're ahead of schedule. I'm just waiting. Yeah.
So I just want to say, I never ever want to give a talk again without Danny going before me.
Yes.
That was an absolutely beautiful introduction. And it's just so important to really understand how metastatic disease is distinct from the primary disease, and how both the science as well as the therapeutic development as well as the clinic really have to think about this differently, which is really where this whole group is all about. I just thanks, Danny.
Yeah, no, and I have to tell you, I think I've learned a lot from Danny, and the analogies that he gives, and while we're waiting for questions to come up, then Peter, that was the whole ultimate goal of today's is to make a very educational day, and my talk will be actually showing folks what we actually, in a very 30,000 foot level of how do we do experiments in the lab, bringing folks who have never been in a research lab, bringing them into the lab. And so I think Danny does always a fantastic job, and I learned, much. But let me ask you a question, Danny, since folks we don't have. So what are your, thoughts? You said very, in a very 30,000 foot level. What are your thoughts on the different races and the populations that you mentioned, right? For inheritability of cancers and metastasis. You touched upon it a little bit, but I know that's one of the things that questions I've gone previously from the audience.
Well, without getting too deep into it,
right.
We know that there are actually differences. I mean, while we're about 99.99% identical as humans, there are differences that exist between different races. And that's part of the reason we have people from Africa who are darker skinned than me, who has no melanin whatsoever. There are other differences that one can look at. And Kent Hunter, a colleague from the NCI, did one of the more amazing studies, in my opinion, where he actually looked in mice, and he's been mapping genes that distinguish strains of mice, just like we have strains of people, and has mapped metastasis modifiers. We've been looking in a part of the cell called mitochondrion. And those are used by evolutionary biologists to define race and actually find certain differences in a single gene, it looks like at this point, that if you have one mutation, it promotes metastasis, and another, it inhibits it. So I saw a chat pop up, but it disappeared real quick.
No, it was from Pat Stieg actually to all of us. Why don't drug companies find drugs for metastasis?
Oh, Pat, well, Pat knows the answer as well or better than I do, but drug companies have limited budgets, even though we all look at them as being highly profitable. To make a drug costs a couple of $2 billion. And metastasis is hard. And I think even showing that one slide where I talked about different kinds of motility, you have 4 different ways just to crawl. And if a cell can't do it one way, it finds another. And that's part of the issue. It's the big word is heterogeneity and adaptability. But the other is the clinical trials for metastasis. I don't know that there's still any great consensus on how to design a clinical trial that specifically targets metastasis. That's because most of the drug companies treat the metastases as identical to the tumor cell, just in a different place. And that is not that's not a good assumption.
Oh, great.
And I'm sure she'll get better answer later today.
Yeah, that's a very great point you bring up. We have one question we'll answer, and then we'll go on to the next just because of time. The question is, can we monitor pre-metastatic niches, and how? Great question.
Yeah, it's a great question, and I don't have a good answer for you, Ting Long. There are people who are doing that in the laboratory. Rosie Kaplan, David Leiden, they're the 2 leaders, I think. So watch for their names. They are working to characterize the pre-metastatic niche. I think it's possible in a mouse or a rat now, but I don't know that it's entirely possible in people yet. But people are working on it.
Okay, great. Yeah, I agree with you. Okay, great. Thank you so much, Danny. And, really appreciate the wonderful, I think, talk that you gave, because it sets up not only my talk but others coming up on the conference. And it's always great to see you. And,
always good to see you. And I apologize to the others because I really would like to be there for everybody else's talk.
I know.
And thanks for the kind comments as well from all of you.
Thanks, Danny. Okay.
Stay safe,
bye.
everybody.
Okay, great. So we're going to go on to now, let me share my screen. So second speaker is myself, and I think Dr. Welch gave a great introductory, on what metastasis is. And like I mentioned, I'm an assistant professor at USC, and my background is in neuroscience, and my research is in brain metastasis. But today, my goal is not to talk to you about brain metastasis, but basically bring you into the research lab, because I know that so many of you are curious about what we do, all of us on the panel, and have never seen a research lab or some of the basic concepts of how we do experiments. So that is going to be my goal for today. So Danny brought up, gave a great lesson on the biology of cancer and metastasis. I'm just going to, not going to be redundant, but just to say, the word cancer, I mean, you got to go back to almost, 400, BC, where Hippocrates noticed that these cells that were not normal had the shape of basically crabs. And that's actually what the word cancer means, is because these cells are shaped like little crabs, and they have, these long feet and hands. Now, with respect to breast cancer, the terminology, we always hear HER2 positive, ER, PR, triple negative. What does that really mean? So the way tumor cells get to function and they work is they communicate with little, antennas on top of their the roof of their cell, so the house. If you think of a cell as a house, then you have antennas, and these antennas could include what are called receptors, and one of those receptors is the HER2 receptor, which is a subtype of breast cancer. With respect to the other subtypes, so ER and PR, these are estrogen and progesterone receptors. These are necessarily not on top of the cells, and I know what I just mentioned, but they're more of antennas within the cell. So think about it as sort of like the Wi-Fi that we have, inside our own homes. And the elusive triple negative, all that means is that that cell does not have either the receptor on its roof, the HER2, or the internal Wi-Fi signals, which is the ER and the PR. And that unfortunately makes it very elusive because we don't have what's called a targeted therapy for it. We don't know how to specifically go after that tumor cell. Danny mentioned this very well. Cancer is more complex. This is, an image of, what the old pathology books and anatomy books showed of what cancer is, that it's just basically two normal cells that start dividing very fast. And we know that that's not the case, that they are not all the same as it is in this picture. And that the word there that I want you to remember is the word heterogeneity. And Danny mentioned this. It's basically very different. So it's like a melting pot of cells, which all come together. So why does cancer kill, or why does treatment fail? And this is the concept, one of the concepts of, tumor dormancy or hibernation. And Cyrus Gajar is going to go through this concept later on, and Peter Kuhn and Alana are going to be speaking after me, and Dolores or Davisio are going to be coming. One of the main concepts is, it goes back to that word heterogeneity. So imagine a tumor cell is different color marbles. You have some purple marbles, you have some red, and you have some yellow marbles. Well, chemotherapy, the primary types of, or the current treatments that we do have right now, they'll kill the majority of the cells. So they'll kill all the purple cells. But what ends up happening is that these red and yellow marbles are basically resistant. They have the various characteristics of becoming resistant, whether they go to sleep, like hibernation, whether they're just very aggressive and they can beat out the chemotherapy. But these are the seeds of what the reoccurrence, or as well the metastasis, metastatic tumor pop-up. These are the seeds for these tumor cells. And to go back to that whole world of heterogeneity, what Danny alluded to is that it's not a simple thing. Danny alluded to the freeway system in LA, or think about it in New York, the subway system, or even Shanghai, the subway system there. And so what you see now is that it's not just the tumor cells that are doing all of the work. They are actually hijacking, our normal body's own, in this case, immune cells or other supportive cells, to help them grow. And so this is just one example from, our research when we do brain metastasis, showing that, tumor cells are sitting here nicely with brain cells, the neurons here. And they're actually, you see what Hippocrates called, the cancer cell, the crab-like. They are basically these little, hand and feet of the cells that are basically holding hands, they're grasping each other. And through these holding of hands, they're passing back and forth molecules that are either good for the tumor cell to grow, or they are setting that environment that Danny mentioned. So metastasis, okay, metastasis is, like Danny mentioned, is just a spread. But what does that physically look like? And this is something that is, I want to make it clear, and Danny did a great job of introducing this. But what ends up happening is that a woman will never, or a man, I should say, will never die of the disease within the breast. It's when those cells pick up and they go through our blood vessels and they start floating around and swimming through the pipeline of the body, and they land up in other organ systems and grow. So for example, when we say breast cancer cells have metastasized to the liver, it's not that the liver itself is cancerous now, it's actually breast tissue growing within the liver. The same concept goes with brain metastasis. So when my neurosurgical colleagues, they take out a brain metastasis from breast cancer, and I know it's somewhat of lunchtime wherever you are, but sorry for the analogy, but the brain is, imagine it has a jello texture and the breast is very fatty. And so it's really when they, in a cottage cheese sector, so when they surgeon goes in into the brain, it's actually jello surrounded, you know, that is, I'm sorry, it's cottage cheese that is surrounded by jello, because that it's breast tissue growing within another area. Another concept is the circulating tumor cell or liquid biopsy, what Peter Kuhn is going to be talking about, is that these tumor cells are now floating around our body. And also, not only are they floating around on our body, tumor cells, whether it's in the primary site, which is the breast, or in other organs, can be sending out these cargoes. And that's the extracellular vesicles what Dr. Davisio is going to be talking about. And these cargoes help promote, like Danny mentioned, the summer homes, of these tumor cells as they're traveling around. And so I took this from the headlines in January, from TMZ. It came out that in January, then this is the misnomer that that we need to basically educate the community about, that breast cancer, when it travels or metastasizes into other regions, it's still breast cancer, but it's growing in different regions. So Taylor Swift's mom, that was the headline, mom also battling brain tumor on top of her breast cancer treatment. Well, this is certainly not the case. It's not that it's that breast cancer, that she has a brain tumor on top of it. It's actually breast cancer that has traveled in other regions. And this also holds true for other cancers, and other types. I mean, we lost two great people this past year, Chadwick Boseman to metastatic colon cancer, and more recently, Ruth Bader Ginsburg to pancreatic cancer. And the term always is metastatic. So these tumor cells, whether it's breast or colon or pancreas, they leave their original site and they go to other places. So the key word is metastasis. Now, how do we look at metastasis in the lab? And I want to bring you into this. And so the way we, everybody imagines, cancer and the research we do, it's sort of like not like Breaking Bad, or it's not like, the things that you see on TV. So, and movies where, we get a DNA sample in the morning and then we unravel everything by at nighttime. This is, research lab. This is how a research lab looks like normally. And so we have, as you'll see, and a lot of my colleagues can attest to, we have our bench tops, we have our different, tools that we use, pipettes, we have all of our, grad students, postdocs, we have Post-its everywhere. And so I want to bring you into what does a research lab really look like, and how do we study cancer cells within a dish? So you've heard of this, studying cancer in a dish. How do we do this? So when, our oncologist colleagues, they take out a tumor, whether it's, breast or lung, we grow them in these little Petri dishes and in the lab. How do we grow things outside of the body? Well, we have to mimic the body, and this is called in vitro or in cell culture. So the way when we get tumor specimens from the OR or biopsies, we work in what's called laminar flow hoods like this. And so we have pictures here. This inside of here, it's sterile, because we want to, our immune system is protecting our body from bacteria and viruses. And we want to study the cancer cells, and we have to do it in a sterile environment so that the tumor, what we are actually studying is actually the tumors and not the fungus and the tumor and the viruses and bacteria that are floating within the air. So we do everything within this sterile environment and are in these what we call cell culture hoods. We grow our cancer cells in what we call media. So this is solution. So this is food for them. Okay, we give them nutrients, and if we're studying, for example, dormancy, we give them less nutrients. Or if we're studying, let's say brain metastasis, we give them nutrients that are similar to the brain environment, the food that our brain receives. And you might say, well, our food is food to our body. No, but what ends up happening is a different environment. So remember that word. Different environments have different nutrients. There's subtle changes that these cancer cells take advantage of. Finally, I said the word mimicking the body. We grow our cancer cells in something called incubators. So this basically it mimics the body's temperature. It mimics the body's carbon dioxide, oxygen levels, And so it's basically a vessel. So if you think about it, you know, it's a vessel for these tumor cells. You know, when we put our Petri dishes in here, they grow in the right oxygenated and temperature, just like the body. Now, how do cancer cells look like? Well, these are 3 different pictures of 3 breast cancer cells that we have. And what's interesting is that they're all breast cancer, but they all look very different. Okay, and this is one of the other hurdles that we have to go through. When we hear breast cancer, we think, oh, all cells look the same. But they're not. They're from 3 different patients. They're all breast cancer, but they look different. And so imagine the genetics that's under there that is driving these cancer cells, how different they are if the cells themselves look by shape different. So how do cancer cells look like? So this is called the time-lapse video. So these are cancer cells just moving over about 2 days worth. We've increased its speed. What you end up seeing is that these tumor cells start what we call proliferate or divide. And you see these little tumor cells here that are floating around. Well, this is sort of them picking up from the dish and just, you know, floating away. And this sort of is what happens with metastasis. So you see this little guy that just came through, and I'll replay the video again. It'll just start floating around. This is, in a very simple way, what metastasis looks like, because these tumor cells are growing very flat and they're holding on to that collagen, for example, that Danny was talking about. And then they'll basically anchor off and they'll start floating around and they'll whiz off. And that's what metastasis is. And you see right here, my little prompter here. So how do these tumor cells, you know, we were talking about the environment, and I'm going to use, you know, one slide for brain metastasis to talk about this. Cells, remember what I told you, they talk back and forth through the antennas, okay? And these antennas basically capture molecules from their surrounding regions or within 2 tumor cells talking back and forth. So imagine if my kids, they were playing back and forth, they're throwing a tennis ball back and forth with each other. That's exactly how our cells communicate within our body, is that they throw little molecules back and forth, and those molecules basically tell each cell what they should be doing. So how do we do some experiments? So we talked about environments. And how do we do these experiments within the laboratory? So remember I showed you the dish here before? We can culture 2 types of cells. Now we have a little insert on top. We can basically culture breast cancer cells, and on the bottom, we can culture brain cells. And so this insert up here is this right here. And we put this guy in this little dish right here. And these little balls here that were floating around are those signaling molecules that the breast cancer cells are basically going back and forth with the brain cells. And we can basically study what those are and how can we stop them from communicating with each other. We can also study metastasis. So remember, how do we study something that is floating through our body? We can study metastasis, what's called on a chip. And this chip is basically analogous to a computer chip. But what we do is we load up these little channels, what we call, with tumor cells. And these tumor cells now whiz through this chip, and based off of how that chip is, it basically can pick up different signals, different molecules. It can pick up different properties of those cells traveling through the channel. And we can test drugs this way. We could test resistance through this. So this is what's called a microfluidic chip, if you've ever heard about it. And it's very small, but that's the most amazing thing, is that you can load it up again with cancer cells and study different properties of it. So let's look under, some very, very simple assays of how do we look at things, in a cancer cell. So going from all the way from its DNA, how that DNA is basically made into RNA, and that RNA is in turn made into proteins. So what kind of techniques do we use? So if you think about, DNA and RNA in cancer biology, is that imagine, an author, you're sitting there, you're typing up a book. If we didn't have word and spell check and autocorrect, you would probably be making these little, spelling errors. These spelling errors are exactly what happens with our DNA or our genetic codes. As cells are dividing normally, sometimes there is error. These errors are now can misspell a word. And if they misspell a word, then the theme of the book or the page is now changed. And that's exactly what happens with cancer cells. I'm sorry, with normal cells. As they're writing their genetic codes, there are spelling errors. And that spelling error all of a sudden turns that normal cell into something it shouldn't be. It changes their storyline and that timeline. And so how do we check the proofreading? We have machines that basically what's called sequencing, and it basically, we can read through the DNA and the RNA and check to see what those spelling errors are. And based off of that, those spelling errors are genes that have changed that normal cell into a cancer cell. And then we could study what those spelling errors are and try to figure out how has that changed the story of that book? And how can we stop that error from occurring or causing more problems within that cell? And so that word is called DNA and RNA sequencing technology. It's just proofreading. The next is, well, how do we measure these errors that happen in a cell? So imagine the gene HER2. We use techniques called qPCR, and we measure basically the RNA of HER2. And so in a normal cell, HER2 or that antenna should be very, it should be expressed at a very modest level. But in tumor cells, it's expressed at much higher levels. And when we quantify this, or we look at this through qPCR. Finally, we get the protein staining. How do we, so for that RNA, for that HER2 to actually work, it has to be changed into protein. It has to be physically changed into the antenna. What we were talking about, whenever we talk about DNA, RNA, it's located in the nucleus here. But that signal, that message has to be changed into protein, and it has to be now shown on top of the cell. We can measure what HER2 signaling looks like by, techniques called immunofluorescence. So it's basically putting an antibody, something that recognizes HER2, and attaching it to the top of that cell, and then that tagging mechanism or that antibody now shines and glows through a fluorescent technology. So for example, when you hear the word immunohistochemistry, or what pathologists look like all the time, is they look at tissue staining. Okay, and the tissue staining, for example, so this is HER2 amongst tumor cells that have been stained for right here. So pathologists, the way they recognize or they diagnose tumors is by staining procedures. So whenever you hear that word, think about this technology, immunohistochemistry. And this is a fluorescence way of looking at it, and this is what we call a non-fluorescence way of looking at it. We can do the same thing to cells individually, so they don't have to be in tissue. So these are basically images that I've taken, and you're looking at cells now. And the green here is the skeleton of the cell, and the red is sort of like the muscle-like, or again the skeleton. The blue in every picture is the nucleus where the DNA is occurring. And so now you can start characterizing the different types of cells. So this is, part of a brain cell. This is a breast cancer cell, and this is another breast cancer cell. And these are basically stem cells looking. And so so you can start looking at the landscape of cells in different ways. Finally, we can make 3D images of those. And so these are brain cells that we have. So these are neurons. We can basically put those pictures together, and you get a 3D formation of cells coming. And this is done through what's called a confocal microscope. We can also separate cells. So we can take cells, for example, in breast cancer. So if you take a tumor mass, you can take cells that are HER2 positive and cells that are HER2 minus. So remember those marbles that I showed you earlier on. If half of those marbles have that HER2 protein on top of them, we can take through what's called the flow cytometer, and we can put these cells through this machine. And based off of if they express that protein or not, they get sorted into one pot versus another. And so that way we can just specifically study this what we call population versus this. And this technology is called fluorescence cytometry. So I hope that you got, you know, a little bit glimpse of what we do in the lab. I mean, this is, again, you know, all of these techniques, there are many, many more techniques, but I wanted to teach you or show you just a little glimpse of what we do, you know, in the lab. And what we call, you know, what the cells like to call us is we are their paparazzi, okay, because we like to glimpse into them. And so with that, I will end my presentation and stop the share. And I hope you've, you know, got a better idea on this. And I will let our next speaker, Alana, go ahead and share, and I will introduce Alana as she is sharing her screen. So I'm really delighted to have, you know, an amazing scientist, you know, Dr. Alana Welch, who's a full professor in the Department of Oncological Sciences at the University of Utah, and an investigator at the Huntsman Cancer Institute, and a co-leader of the Cell Response and Regulation Program at Huntsman Cancer Institute. And Dr. Welch's laboratory studies, of course, breast cancer metastasis. Dr. Welch completed her PhD in cell and molecular biology at Baylor College of Medicine. And then she joined and did her postdoctoral training with Michael Bishops and Dr. Michael Bishops at UCSF. Dr. Welch started her laboratory at Huntsman in 2007 and was granted full professorship in 2019. So congratulations, really, Alana. Dr. Welch's laboratory is focused on in vivo, so animal modeling of human and mouse breast cancers. And Dr. Welch's group has discovered pathways that are really, really important for looking at that interaction of cancer cells and immune cells called macrophages. So Alana, please go ahead.
Okay, thank you very much. Just double checking you can hear me. Yes?
Yep,
okay, great.
You're good.
So yeah, thank you for that nice introduction, and I'm really happy to be here. So thanks to all the organizers for putting this together. This is really a special event, and one of the silver linings, I think, of the COVID crisis is that we can still communicate and interact. So I'm here in Huntsman Cancer Institute, but up against the foothills of the Wasatch Mountains in Utah. And my lab actually works on two different things. One is more about how cancer cells metastasize and how they survive in distant sites and what their interaction with the immune system is, and particularly macrophages, as Josh already mentioned. But today I decided to talk about the other half of the lab, which is really about sort of functional precision medicine in breast cancer. And what is it that we can do right now and be creative with new models of metastatic breast cancer to hopefully affect treatment now while we're still trying to figure out more about the hows and whys of breast cancer. So I like to start with this slide because when I first saw this data, it was so shocking to me. And so what you're looking at are data from the World Health Organization, the International Agency for Research on Cancer, looking at breast cancer across the world. And the blue lines that you see here are on diagnosis of breast cancer. So that's the incidence of the disease across the world. And the red lines is representing mortality. So the amazing thing to me is that, well, the first thing you notice is that in the more developed countries, shown here on the top, we have more breast cancer. We have more breast cancer diagnosis. And that is actually mostly due to the fact that we screen for breast cancer using mammography, et cetera, is a very well-known way and very effective way to detect breast cancer. But what I think is most striking is that the red lines, the mortality, and this is the number of people who die every year relative to the population in each of these countries, is strikingly similar across the world, whether you're in a developed region or a less developed region of the world. So what that tells me as a scientist is that we still don't understand metastatic breast cancer, because deaths from breast cancer, as you've already heard, are actually due to the metastatic form of the disease. And so there must be something about the biology of metastatic breast cancer that we're not capturing by all this increased diagnosis. A deadly breast cancer is a deadly breast cancer no matter where you're living, and even access to our most advanced therapies in the developed world are not enough to really impact mortality the way we want them to. So the questions that we're trying to ask is, can we predict which of these so-called blue breast cancers that get diagnosed at high incidence in the US, for example, which of those is going to recur as a deadly metastatic tumor? And the reason why that question is important is because maybe you can do more for that population without necessarily over-treating the population that will not recur. And secondly, and what this talk is focused on, is could more individualized therapy for each of these patients reduce mortality? Can we come up with more personalized therapies? The reason why that's important is shown on this slide. So on the left is what you're probably all already familiar with, which is a typical course of breast cancer diagnosis, therapy, which encompasses surgery with some kind of systemic therapy, either before surgery, which is called neoadjuvant therapy, or after surgery, adjuvant therapy. And the whole point of this therapy, of course, is to prevent metastasis, because as Josh already mentioned, that's why most people, and Danny too, why most people die of breast cancer. So what happens is adjuvant therapy is given after surgery, so the tumor is removed, and yet you're still wanting to treat any cancer cells that may have already escaped from the tumor. So this is why it's called adjuvant therapy. But despite all of our best efforts, we have 20 to 30% of cases will recur eventually as distant metastases. And that leads to, as you know, more than 40,000 deaths every year. That's just in the US alone. Now, we've already gone over the different subtypes of breast cancer. And what I want to highlight here is we have a lot of therapies approved in breast cancer and in advanced breast cancer or metastatic disease. In fact, I would argue we almost have too many therapies. And we actually, in the metastatic setting, the choice of a therapy is often really based on trial and error or convenience, oral drugs versus IV drugs, et cetera. And while they are tailored according to these clinical tumor subtypes, I think we might be able to do better to personalize the therapy with the drugs that we already have. So that's what this project is based on. And of course, there are people doing precision medicine in breast cancer. And I would say that actually breast cancer is probably one of the first cancers in which some form of precision medicine was used, in which, for example, if you're estrogen receptor positive tumor, you will get hormone therapy. That's some type of precision. But nowadays, most people would consider precision medicine doing DNA sequencing on the tumor in order to find mutations and then target, if possible, those mutations with a drug that might make a personalized therapy for that patient. However, what we're trying to do is actually incorporate functional responses, and I'll get to that in just a moment. So if we're successful in doing so, theoretically, the impact would be you could choose therapies that might have better efficacy, so have better outcomes for these patients, but also I think it's very important to point out less toxicity, because you could not choose therapies that are going to be ineffective and spare the toxicities in those patients. So what is it we're doing? What we have developed in the lab, and others are doing similar work, is to take tumors from patients, whether those are from the breast or from a metastatic site, such as liver or lung. And then we originally started by engrafting these into immune deficient mice. So they're growing as xenografts, which is one species into another. And there we can study these tumors. And I'm not going to go into great detail about these, except to say that the tumors are pretty amazing in how well they retain their biology while growing in these mice. And then we also can grow these in sort of three-dimensional gels in the lab called organoids, where they grow in a microenvironment that's sort of similar to the breast. It's obviously not perfect, and it's not in a living organism, but we still can do quite a bit with that. And the idea is, can we do this co-clinically? So can we do this at the same time that a patient is undergoing her standard care? And then can we use these models to learn more about the tumor? So in the first place, you can do analysis of the models that come out of these cancer patients. And you can ask questions like, how well do the models represent the cancer that it came from? And we do this oftentimes by looking at genomics, such as DNA mutations or gene expression. But I would say that this really just gives you a snapshot of that tumor. So a single point in time, this is what it looks like. And we can compare that back to a single point of time in the patient as well. But then we want to take it another step further, which is, can we get functional assessments to answer questions like, how does the tumor actually behave over time? Because since these are growing in living animal or as a living culture in the lab, then we can ask questions like, well, which drugs does it respond to? How does the tumor evolve over time? And we think that this part is really, really important because it's more than a snapshot. And using the two things together, we can then document the fidelity between the models that we create and the patient. And now where we're moving is actually, now that we're convinced that they are high fidelity models, is moving now toward, can we return those results to the clinic? And that's what I'll be talking about here. So just a summary of the models that we've collected or generated so far. This is actually under review right now. And we've really made an effort to try to get the models made for breast cancers where that's the greatest unmet medical needs. So in other words, metastatic breast cancer that are recurrent over time and in the face of therapy. So we have models that represent pleural effusions. This is the fluid that fills up around the lungs when there's lung metastasis. We have ascites from the peritoneum. This happens a lot when there's liver metastasis. We have brain mets, we have bone mets, we have skin mets, we have spine mets. We have breast tumors that have come back in the breast after therapy. And most importantly, I think we have tumors that come from the same patient over time. So for example, a primary and metastatic pair from the same patient. And this allows us to try to get at that question that Danny brought up, which is, how is the metastasis different from the primary? And then use the model for the metastatic tumor as the one to try to determine best therapy. So the advantages of doing this, of these models, is that they are pretty good representation of human tumors. They're not perfect, of course. A model is always a model, but they are pretty good. And one important point is that the ER-positive tumors do maintain their hormone sensitivity and resistance. And this was a need in the field for a long time because standard breast cancer cell lines are not necessarily, there's not very many of them that are hormone dependent, and traditional genetically engineered mouse models typically are not estrogen dependent. So that was an important advance. The other thing that was really interesting is when we make these in the mice, they actually do metastasize in the mice to organs that are kind of mirror what happens in the patient. So that allows us models to study that process. There are disadvantages as well. So there are biological disadvantages. So we can't study anything to do with the immune response in these mouse models, because as I mentioned, the mouse has to be immune deficient in order to allow the human tumors to grow. And so that's a definite caveat. And we're working on putting human immune systems into these mice as well. But I won't have time to talk about that today. The other disadvantages are just, this is very labor intensive, it requires live animals, and it's expensive. It's also, if you want to test drugs in these PDX models, it's really not feasible to do large scale drug screens. It's better for validation. If you have a hypothesis and want to test it, then it's okay for that. So how do we overcome these disadvantages? Well, we decided to try to make all of our PDX models into these three-dimensional organoids, shown here, which we call PDX organoids or PDXO. And these are the ones growing in three-dimensional gels in the lab. So we've been successful at making organoids for pretty much all of these models. We have a few that have failed, and those are shown in red, and some that are still in progress. And of course, our collection is growing all the time. The advantage of doing this is that now you're growing it in the lab in dishes, and so you can actually do large-scale screening. You can do genetic screens, you can do drug screens, and then you can also ask about, well, what happens when I add this cell to the culture or this growth factor or whatever. So you can do biological experiments with these lines in a much easier and more cost-effective way. So the long-term goal here is to generate paired PDX and organoid models from 100 different breast cancer patients. We're about halfway there. We have about 60 or so now. We characterize all of them by genomics, so we know which mutations are present in each model and gene expression profiles, et cetera. And then we screen our organoids for drug sensitivity. And we do this with already existing drugs that are FDA approved for breast cancer. We also test FDA approved drugs for other cancers, as well as experimental drugs, because ultimately this is a research project as well. And the idea here is when you screen drugs across 100 breast cancers, then hopefully using this genomics and other information about the models, hopefully you can come up with biomarkers of response or resistance for each of the drugs that look like they're promising. That way, when you design a clinical trial, you can design it in a way that can test the right population of cancers for that drug. And the way we do this is by testing each drug with 8 doses each, so that we can basically have one library of drug, and we can do this in it's kind of a medium throughput fashion where we test 384 at a time. And we do that in quadruplicates, so with 4 replicates for each one, just to be sure that we're getting accurate results. And we've also run our screens at different time points over passage. So the question of, well, how long can you keep this cancer in the gel? Can you keep it for a year and you still get the same results of the drug test? The answer is yes, so that's good. So ultimately the result here is hopefully to tell us which tumors respond to which drugs, and with more research eventually why. We want to understand that mechanistic question. So the type of results we get back are sort of shown up here as one example. This is a drug called birinapant. It's still an experimental therapy. And what we see is this is the highest concentration of drug up here down to the lowest. And then we're testing different patient models across the bottom. So when you see a pattern of red, that means that this drug killed that person's tumor quite effectively, all the way down to a pretty low concentration, whereas it didn't even touch this person's tumor. So we're looking for those kinds of patterns. And when we basically combine all this data, this is from an initial pilot screen across about 16 patient tumors and 40 or so drugs, what you can see is there's a lot of variability. This is one of the problems we have in breast cancer, is there's such heterogeneity that that's why not all cancer drugs work for all patients. And we have to figure this out. So that's what we're trying to get at. So what this is showing is the most effective drugs for each model are the darker color. So when there's black, that means it really killed everything. And then the darker purple means the drug was more effective. The yellow means those cancers are perfectly happy growing in the presence of that drug. And I'm not going to go into a lot of detail about the patterns that we saw here, but I'll show you one example, again, with this drug birinapant. So what we can do is we can rank the effectiveness of this drug across different patient models. So the patient HCI15 had the best response to this drug, where there was complete killing, whereas HCI17 had no response, and so it's bright yellow. So we can rank these. And then we can start to kind of guess, okay, what level, which ones are going to be resistant and which ones are going to be responsive. And I won't go into the details, but it's a mathematical formula that helps us figure this out. And what you can see with birinapant is that most of the tumors that are responding to this drug are marked over here in red, which means they're triple negative breast cancers or basal-like breast cancers. But not every triple negative or basal-like breast cancer is actually responsive. So that tells us, this is interesting, and it might indicate there's a subset of TNBCs that could benefit from this drug. Now, of course, this data all comes from organoids, but what about in vivo? Can we actually shrink the tumor in a mouse? And so this is just shown here. It's all color-coded. And what you can see is that the ones that we predict to be resistant, shown here in orange, black, and pink, are in fact growing perfectly fine in the mouse, even in the presence of the drug over time. Whereas the ones that are up here in blue and red are the ones that are really sensitive to the drug. And we actually shrink those tumors away, in some cases all the way completely gone. So this indicates for us that converting our mouse models into these organoids might be a very feasible way to ask questions about which drugs might be effective. And now, of course, we want to understand what are the markers of ones that are sensitive or resistant to this drug so that we can design a clinical trial. So we think that these approaches will help identify new breast cancer drugs for the future, but the big question is, what can we do right now? We really keep this in mind. And I'm going to just switch gears really quick for the second part to tell you about our precision medicine trials that we have going on right now that I'm very excited about. Now, the reason these trials came about was because of retrospective data. So when we were first making our PDX models and growing people's tumors in mice, starting from 2007 until we published it in 2011, we made the accidental observation that everyone whose tumors actually grew in the mice died of their cancer. And in contrast, people whose tumors we couldn't grow in the mice were perfectly fine. They were disease-free. And so this got us thinking, like, is there something special about tumors that can grow in mice? And we sort of jokingly call this metastasis to the mouse. It's sort of a biological readout of how aggressive the cancer is. But all of this was on retrospective data. That means we had the data, we saw the pattern, but we were looking back in time to say okay, this one grew, what happened to that patient? And so then we instead started a prospective study to see if we could validate this result, if it was really real. And so what we did is we started the study called Towards Functional Precision for Breast Cancer. It's a clinical study that enrolled newly diagnosed ER/PR negative stage 2 and 3 breast cancer patients. So these are people who are at relatively high risk of recurrence within the timeline of the trial, which is about 3 years. We collect a biopsy of their primary tumor before they start treatment. So these patients are going to undergo neoadjuvant chemotherapy prior to having their surgery. And we ask, does the tumor grow in the mouse or not? And the hypothesis is that the ones whose tumors grow in the mouse would have poor outcome. In other words, they would have metastatic recurrence. And in our hands so far, this trial is still open, and we're going to present an interim analysis at San Antonio this year. But we've have a little over 70 patients on study now. Of those, it's about 25% whose tumor does grow in the mice. And when we do the analysis of the subset of patients that have been on study long enough to look for time to recurrence, we see a very significant correlation of PDX engraftment with relapse. In fact, the odds ratio is 20. That means people whose tumors grow in the mouse have a 20 times higher chance of recurrence. Now, the current clinical standard for predicting recurrence in triple-negative breast cancer is actually whether or not the patient has residual cancer in the breast or lymph nodes at the time of surgery. So they've gone through their chemo, and if there's still cancer left over, then that is a poor prognostic factor. And that's called pathological complete response. So when we look in our study, again, this is a small number of people, we're not seeing at this time any significant stratification of these patients by whether or not they had a complete pathological response to chemotherapy or not. However, if we stratify them by whether or not their tumor grew in the mouse, you can see this really striking effect. So people whose tumors can't grow in the mouse, they have not recurred yet at around 3 years. But the people whose tumors do grow, unfortunately, have these really high frequency of early recurrences. And this is looking at overall survival, so that they're actually not only just recurring, but they're unfortunately dying of their disease. And this is looking at relapse, so just the recurrence. So we can really significantly predict this. But that brings me to the next point. If we have their tumor growing and we know that they're at high risk of recurrence, why don't we do something about it? So what we're doing now is using their models, whether it's the mice or the organoids in culture, to screen drugs. And the idea is then, can we return these results back to their physician in the context of a tumor board that might be able to hopefully inform their therapy for the better. So I'm going to take you through one example. This is a patient on our trial called we call her TOE 19. She's the 19th person enrolled on the trial. She was diagnosed with triple-negative breast cancer. We got the biopsy and made the PDX, engrafted in the mouse. We had that established in 90 days, which means it was a pretty aggressive tumor. In the meantime, the patient underwent neoadjuvant chemotherapy, standard of care. At the time of surgery, she actually had a pathological complete response, so no detectable cancer, which normally would be a good prognostic outcome. However, because her PDX was growing, and we made organoids from it as well, that's the PDX-O, we had we would hypothesize that she would be at high risk of recurrence. However, we froze her organoids actually because she had a path CR. So this is before we really knew how strong our data was, I guess. And we did some genomics on the tumors. And what we found was that this tumor was an interesting type called a luminal androgen receptor subtype. And really all this means is it's a triple negative breast cancer, but it's expressing the androgen receptor instead of the estrogen receptor, which drives a more ER-like phenotype, even though there's no ER. But these kinds of cancers are actually sometimes thought to be good candidates for androgen receptor therapy, such as used in prostate cancer, like enzalutamide. So that's what we learned by doing the genomics analysis on the tumor. Unfortunately, we were correct in that this patient would have recurrence, and less than a year after her surgery, despite the complete response, she had recurrence in the liver. Immediately upon hearing that, we thawed out the organoids and we started running drug screens. And then we validated those drug screens in the mice to see if they really could have effect in a living model. In the meantime, the patient went on standard care, first-line metastatic therapy of capecitabine, but unfortunately that was not very effective for her. What did we find when we did our drug screens? Well, we found that the gray arrows show sort of her standard of care therapies. So the first two are part of her neoadjuvant care, therapy, the doxorubicin and the paclitaxel. And you can see those were pretty effective. So it's toward the dark end of this graph. And you can see that just like in the breast, the tumor shrunk away completely in the patient. These drugs were relatively effective. I will say in our mice, they didn't completely cure the tumors. So that is consistent with the fact the patient came back with recurrence. What we were most interested in though is this red arrow, which is a drug called eribulin, which is approved. It is FDA approved in breast cancer, but it's not usually used until like the third line of treatment in the metastatic setting because it's quite expensive. And so we tested this one in the mice. We also chose a couple others that were kind of intermediately effective in our organoids. The reason we chose these two, talazoparib and cabozantinib, are because there were clinical trials available at our institute at the time for those drugs. So we thought we'd check how those worked. So the results in the mice are shown over here. So eribulin completely cured all the mice of their tumors. The talazoparib and cabozantinib were moderately effective, so the mice basically had stable disease. We also tested enzalutamide, the prostate cancer drug, because of the genomics findings, and we saw that it did slow the growth of the tumor, but the tumor is still growing. So that suggests that getting information just from genomics was not enough in this case to find the most effective therapy. So back to our patient, we returned our organoid drug screening results at this point in the timeline. Because the patient's tumor was actually positive for an immune marker, she went on an immunotherapy trial that was available at the institution that was the combination with cabozantinib. Unfortunately, the tumor still progressed on that trial. And at that point, we had already the mouse results showing basically complete cure of the tumors in the mice with eribulin. So the patient was actually put on eribulin. And amazingly, she had a complete response of her liver mets. They completely went away. And she stayed stable like that for quite some time. Unfortunately, what happened though is she got a new bone met. So even though the liver mets had completely disappeared, there was a new bone met that occurred on therapy. And so the therapy, the systemic therapy of eribulin was stopped, and she underwent radiation therapy to the bone for the bone met. And eventually off treatment, that the liver mets came back. And at that time, she was given a different drug, carboplatin, which again, based on our mouse and our organoid data, would not be predicted to be especially effective. And unfortunately, the patient progressed and eventually died of her disease. One of the things that we were able to do in this case was to generate another set of models from the metastatic disease using the ascites when she had the liver mets prior to the eribulin treatment. And these models all showed the same thing, that eribulin was the most effective therapy in this case. So the questions that remain, so we don't know if this new bone met that popped up was ever represented in our original models because of the heterogeneity. That's like the marbles Josh was talking about. Maybe those marbles were hidden in the body but weren't actually captured when we made these models. And maybe that's why they grew on eribulin. We also don't know whether the liver disease that came back after therapy or off therapy would have responded again to the same therapy or not. Generally, clinically, when you give a systemic therapy and you have progression, it's considered that the whole disease in the body is resistant, but we actually don't know that. So these are the questions we would like to answer. And as a result, we have now opened a new trial, or just about to open a new trial called 4C, in which we're doing all of the same work, but all in the metastatic setting. So rather than starting way back here, we're going to enroll people who already have metastatic disease, and we hope that we can have impact in that arena. The big questions here are, the goals are really to develop the infrastructure to do this in the metastatic disease. Getting these metastatic biopsies is not easy. Growing the models, doing the drug screen, et cetera. And we want to do this in a fast turnaround. So we can do this between scans, so within 12 weeks, so that a treatment could be adjusted if needed. And the other goal is to build clinician confidence to act on this information. I don't think anyone else so far has done this in breast cancer. And the question is, will they believe our data? Will it be meaningful in the clinic? And that's one of the feasibility goals of this study. So 4C is basically designed as soon as someone relapses with distant metastasis, we get a biopsy. We do our thing that I already talked to you about. In the meantime, the patient will go on standard of care therapy for the first line. When we return these results back to the clinic, then the physician will make a decision to keep that therapy or switch to a different one. So with that, I'm going to wrap up. I just want to thank everybody in the labs who have contributed to this project. For this particular project, they're shown here in bold. We also have a fantastic collaboration with Mike Lewis at Baylor to get even more models. We thank our patients for donating their tissue and to allow us to make these models, but also the physicians that we're working with to develop these trials. And these are all the funders that contributed to the project I talked about today. We're also grateful for our common funding, which actually is for a different part of the lab that I didn't have a chance to talk about today. But with that, I'm happy to answer questions in the panel discussion, and we can move to the next speaker.
Thank you, Ilana. Wow, what a great example of the, precision medicine and how, the parallels work. And there are several questions, but we'll move to the, we'll have them during the panel discussion. So while Peter puts up his slides, I'm going to introduce him. So Dr. Peter Kuhn is the Dean's Professor of Biological Sciences and Biomedical Engineering and Aerospace and Mechanical Engineering here at USC. He's also the founder and the co-founder of the USC Michelson Center for Convergent Biosciences. Peter, by training, is a physicist, and this is exactly what, thinking outside the box and bringing people from who aren't necessarily started their careers looking at cancer, but how they can contribute. So his research is, has shed light on how cancer spread through the body, like, I mentioned through the liquid biopsies. He's published over 200 peer-reviewed scientific articles and patents as a result, and he founded Epic Sciences in 2009, which helps develop cancer diagnostic products. And today, Epic is one of the key partners with most biopharmaceutical companies in developing precision ways for diagnosing cancer. So, Peter, looking forward to your talk. It's all yours.
Great. Thank you, Josh. So as I was listening to the last 3 talks, of course, I rapidly reconfigured my talk because this is a fantastic flow of information, and I hope that everybody in the audience is sort of really enjoying this tour as we're going through this. So I will talk about the liquid biopsy as such, and really from the perspective of just picking up from where Elena just left off, of how do we actually implement precision medicine, bridging the gap to enable that continuity of care as the cancer evolves over time. So a lot of this work, of course, is now happening also in the context of post-outbreak, and I just wanted to mention that. because it is actually important, because it is where a lot of the technologies that we have all built over those past number of years are becoming even more front and center. It is now more clear than ever, I think, that all the work that we have to do in cancer research, it really has to be patient-centric. But it also has to be remote enabled, as we have seen through the various aspects, what has happened during the pandemic that has led up to now. And a lot of it has to do with limited access to healthcare facilities, etc. So home health is becoming more and more important. And as we are thinking about liquid biopsies, of course, you can think of mobile phlebotomies as well. And oral drugs being delivered through the mail, as opposed to certain aspects happening in the clinical setting itself. And that really then allows us to help the patient to be prepared and make best use of the interaction with the treating physician in the clinic itself, which is something that we have always talked about, we have always envisioned. It's now just becoming much more front and center. There is this whole discussion around the Internet of Things as it relates to health, and what all we can measure in terms of the health performance of an individual patient. So I will try to wrap this by starting with the patient, talking about the disease itself, the characterization of the disease with the liquid biopsy, and then bring it back with the very last slide, essentially, to really point to this importance of this interplay between the disease and the patient itself. So this is a patient who has really shaped our thought process from very, very early on. This is a lung cancer example. So, as you can see, that PET CT scan showed a singular nodule in her lungs. And so the decision, because of her frailty, the decision was made to treat this with stereotactic radiosurgery at the time. And when she came back 3 months later for her next imaging session, that primary tumor indeed responded very nicely to radiotherapy. But of course, now she had bone metastasis and mediastinal metastasis. So there's clearly something that we overlooked in this process along the way here. And and she was one of the patients who was early on in one of our liquid biopsy trials, and she had a very large number of tumor cells circulating in her blood at the time of that first at the beginning of her stereotactic radiosurgery. So in hindsight, one has to say, well, wait a minute. In a patient like hers, she would have actually been better off with systemic chemotherapy or palliative care, given her frailty and her age. So this is really then something that makes us thoughtful, right? It gives us pause. It tells us, well, wait a minute, how do we actually go about measuring the disease in the human body as we go forward? So the next couple of slides are really meant to just set that stage. So to start with, what is a liquid biopsy? So it's a biospecimen. It's a blood sample. It's a blood sample. For the most part, we're thinking of this as a peripheral blood sample, but can be from various blood sampling throughout the body. It can also be a bone marrow aspirate. It can be a CSF sample. It can be an ascites. Any liquid sample is really something that we call a liquid biopsy. So it's a biospecimen. In each of these biospecimens, there is a heterogeneous mixture of cellular and non-cellular components. So if we think of the blood itself, right, so you have your immune cells in there, the white blood cells, right? You have your red blood cells in there, and then you have all of the plasma, so all of the proteins and the small molecules, the chemicals that flow through the body that give you energy, nutrition, and provide for that defense mechanism. And amongst those, you have, in that whole mixture, you have a small set of very specific cells that we're interested in, right? Those are the cancer cells, or the cells that come from the tumor microenvironment, the proteins that we can measure that are either a direct result of the cancer or an immune response to that cancer. And of course, the nucleotides, meaning the DNA that comes from the cancer itself, that we then try to measure. Now, of course, and it's interesting to just reflect back and ask the question of like, well, where does the science come from, and where are we at today in 2020? So this was actually first discovered by Tom Ashworth in 1868. So it's an old observation during an autopsy where a pathologist recognized that there were cells that in fact looked identical to those from the primary disease were first discovered. And it's that type of a fundamental discovery that really started this whole process of science and this journey of discovery to understand how does the disease evolve in the human body itself, and then how can we actually make use of that. And you know, I used to, when I entered this field, I used to say, we still have not reduced this to practice, but again, thankfully in 2020, we can actually say that we have finally reducing this to practice, and this is becoming useful. But why has it taken so long? So if we just think about, and many of you who are either patients who have been diagnosed or have been diagnosed with cancer at some point, or just have gone to see your doctor for your annual physical, you do blood workup, right? So, and in the blood itself, you then get your white blood cell count. That's typically a couple of thousand per microliters. You get your red blood cell count, which is a couple of million per microliters. And then there's a platelet count in there as well. Now, these white blood cells, so the immune cells, right, they split up into 5 big subtypes. And those are, again, counted in tens to hundreds to thousands per microliters. But the circulating tumor cells that we're now trying to find, they are in the single digits per milliliter. Or even if we look for DNA from the tumor, we're looking at the same level. We're looking at that one copy that reflects that one cell per milliliter of blood, right? So we are now upping the bar in terms of what we have to look for by about 1,000-fold. And that is what makes this just so difficult. And then, of course, these cancer cells, they know how to adapt. So they don't look super different from the immune cells. So for that, it essentially becomes the way we like to think about this is as an extremely fast, high performance computational game of Where is Waldo. So when you think about this, so this is meant to depict a microscope slide where we would pack about 3 million cells onto here. And the question is, within the 3 million cells that are on here, can we find that 1, 2, or 3 cancer cells, right? Can we actually find Waldo? And the tricky bit about finding Waldo is, of course, that there are lots of either things or others that look just like Waldo, but those are false positives. So the way we have to train up our algorithms, our approaches, is really to separate them out as well as possible. So that's one big challenge. The other big challenge that we always have to be aware of is we have to ask the right question early on. So us as scientists, oftentimes, you know, we fall in love with our tools, we fall in love with our technology because it's really cool. It took us forever to build it. It's very elegant. But I think what we have learned by working with our patients for many years now is really to right away in the beginning of a research project, ask this question about why. What is that clinical context? And I love this example of the caterpillar and the butterfly, because if I ask the question of like, well, which one can fly? Okay. In fact, if I would sequence their genome, their DNA is in fact identical. So that will not answer that question. But I have to look at that proteome, which is what Elena said, the functional readout, right? So that is where those aspects connect, right? So the proteome, of course, will tell me which one can fly. Now, if there would be a pathologist in the room, the pathologist would start laughing and be like, well, Peter, you can also just look at the pictures up here. The one with the wings is likely the one who can actually fly. Okay, so that's why it's always important that we ask this question of what is that clinical context of use? So with that, we get to this, what is the liquid biopsy in 2020? Because we're not anymore in 2005 or 2008 when some of us entered this space, right? So we are now at this level of what we call a 3rd generation liquid biopsy. In this 3rd generation liquid biopsy, we recognize that a tube of blood is just really rich of information. We can find circulating tumor cells. In fact, we can find circulating cells of the tumor microenvironment, which again give us a signal of what's actually going on inside of that tumor. We can find oncosomes, and Dolores, who will talk after me, will talk about these oncosomes as well. We can then, from these CTCs, we can actually sequence single cells, so we can do the genomic information of single CTCs and of the nucleotides that are floating around, that cell-free DNA that's floating around, we can figure out which is normal cell-free DNA and which is tumor-derived DNA. And now we can do all of this within a single tube of blood. And so that now opens up a whole wide array of opportunities to really participate in clinical care. Now, again, in the beginning, in this research use only framework, but with that goal of advancing it as fast as possible into clinical care, exactly what would be that next step of what Elena just said about her ongoing trial as well. How do we take research? How do we take research forward? So this is just how this workflow essentially works, right? So whether it is a tube of blood, or it is, in fact, we do primary biopsies as well, where we touch that tumor onto glass slides, but we also use bone marrow aspirates, CSF, etc. We then have developed a very standardized approach because we want to compare these different samples. And that's actually a really hard thing to do. If I take a sample out of different parts of the body and try to compare them, put them on the same playing field. So we do that, we cryobank that so we can run along with clinical trials, and then we can do a whole bunch of analysis with that allows us to get to the genomics, which is really the blueprint, but also to the proteomics on the right-hand side, which is that functional readout, right? For those of you who live in the Bay Area, think of the Millennium Tower in San Francisco. That's always I love that example because its blueprint actually looks pretty much okay. It's really hard to find a mistake in that blueprint, but in fact, it's leaning, right? So its functional readout is actually where you're like, oh, whoa, wait a minute, there's something wrong with the Millennium Tower, okay? So that's why we like that interplay between genome and proteome to really allow us to make progress. I just want to show one example that is really a really important way to think through this. So this is a patient, she was diagnosed in 2012 with primary breast cancer with metastasis to the bone. And she enrolled with us on a clinical trial in 2013, so 7 years ago. But then for the next 4 years, we followed her along this journey of the disease under therapeutic pressure. She had 8 different approaches to a therapeutic intervention. And at some point in 2014, developed liver metastasis. And when we looked at her liquid biopsy results, and what I'm showing in this first slide here is to simply say, oh, okay, interesting, her CTC counts were actually consistent with her other biomarkers. So that was for us a place to be like, okay, this all seems to work. But how do we characterize the disease, right? How do we relate what we are seeing here, not just in saying, you know, disease progression, disease response, but how do we characterize this so that we can relate the treatment modalities to what we actually see in the cancer itself? And this is what we really did here. So for starters, what I just mentioned is that the CTCs as well as the ctDNA, the circulating tumor DNA fraction correlates very nicely with the traditional biomarkers. That was just important to know. But then, of course, we also realized that how interesting her genomic profiles, which we call copy number variation profiles, really inform us about the lineage, the ancestry of her cancer. And so we have done over 500 genomes in her alone. And what we saw in her is actually that she had a pretty stable cancer genome with very few variations, which is why she actually, despite the fairly heavy burden of metastatic disease, actually survived for a long time. It responded to these various therapies. The CTCs that we found early on matched the genomic profile from the tissue biopsies. And interestingly enough, actually different CTCs we were able to associate with either that primary breast cancer mixed with the bone metastasis, but the clone that really showed up in the liver is actually a distinct clone. And we saw that much earlier than the actual liver metastasis was found that was specific to that liver metastasis. These clones are stable over time. After 3.5 years, here on that right-hand side, a new clone showed up. And that is when really, when therapy failed and that clinical escape happened and the patient passed. And this was really tricky. And we're trying to figure out what was going on, and I'll come to that in a minute. The cell-free DNA is because it's a bulk measurement, right? So it's a representation of the aggregate of the most abundant circulating tumor cell clone, which again makes total sense. The ESR1 mutations. So this is really a key, a key readout to understand whether or not ER-targeted therapy works or does not work anymore. Showed up only after that first year, so that did not exist early on. The P53 mutation, again, really interesting, right? Because that tells us really about the genomic stability and the stability of this replication cycle and process. These mutations showed up first, again, now in 2015, so 3 years after the disease was originally diagnosed. And with that, all in all, it was super interesting that the single mutations in that particular cancer were actually quite rare. So her disease was really, for the most part, driven by these large-scale copy number variation changes. And again, that was important to understand how that disease actually progresses over time, and that these single mutations were rare, but then important to start recognizing. The oh, I thought there was one more. Sorry, I forgot there's one more animation that's somehow missing. So this new clone after 3.5 years actually carried a Pi3 kinase mutation, and so at the time, neither the Novartis Pi3 kinase drug was approved, nor the Roche Pi3 kinase drug was approved. So one would imagine in hindsight, right, that if we would have done this in real time, there would have actually been another therapeutic opportunity for that patient, and to see what the disease would have done under that therapy approach. So this is really one of those examples of how we envision how we can manage individual patients over time with the liquid biopsy, because obviously you cannot do primary biopsies 17 times over that time period. And also, obviously, somewhat in quotes, is that because of the heterogeneity of the disease, you really have to think of the complementarity between the primary biopsy and the liquid biopsy as we go forward. There's of course a question of how we translate this, and Josh mentioned this in the beginning, in the introduction, is that this is work by Dr. Howard Scher from Memorial Sloan Kettering Cancer Center in prostate cancer, where he then worked with Epic Sciences, so the company that licensed the technology from the lab, and actually half the lab went over to the startup company at the time when we did that. We take this very serious, right? Because in order to bring these kinds of tools and technologies to the patient, we have to figure out how to commercialize them, how to put them out into companies that can make then that investment to run these large trials that are necessary to demonstrate the benefit. And in this particular case, there is a variation of the androgen receptor. And again, Elena mentioned that androgen receptor that we believe actually plays a role in breast cancer as well, but this is all in prostate cancer, of course. So, and as long as you don't have this variant, which is called androgen receptor variant 7, as long as you're negative for that, you will actually respond quite well to androgen receptor targeted therapy. But if you have this variant number 7, so if you're positive for that, as you can see here, these patients, therapy fails, and their survival is really very, very limited. Now that's important to know, but the question is there something that we can actually do about it? And in this particular case, actually, if these patients switch from that targeted therapy over to taxanes, to chemotherapy, they will actually have a much higher chance of surviving, out to an extra 200 days. So it's an important way to really think about how we do this in real time over the course of the disease. So for us, the question now is really in this whole science framework, it's just like, how fast can we repeat this, right? We have now done this in research. We have done this in moving it through into product, moving this into clinical care, because we have to do this for every decision point in cancer as we go forward. So we have built up this entire framework at USC within this Convergent Science Institute in Cancer that really brings two major components together. One is that comprehensive cancer profile with the 3rd generation liquid biopsy approach, together with a whole lot of mathematical oncology, because it's really the mathematicians that then help us bring this data together. And so the mathematics has really become an integral part how we think about the disease as we go forward. Again, as I said, we have to really, really keep in mind, and that's a whole other lecture. Maybe we do that in a future session like this, where we talk about how the health and the health performance of the patient really plays in with all of this. But that's an important element to this. So with that, I just wanted to finish and say, digitizing the cancer, which is where scientific groups like ours really come into play, because that's where our training is really from. So digitizing the cancer as well as digitizing the health is we believe, incredibly important to deconvolute these healthcare disparities. And we have to do cancer research in the context of treatment pressure as well as human individuality to really change the game as we go forward. So with that, of course, a huge thank you to those who are collaborators and funders of the work that we do. And with that, I will finish and hand this back to Josh.
Thanks, Peter. That was great and fascinating. There's a lot of questions coming through, and we'll ask them in just a couple of minutes. Last but certainly not least, we have our last speaker for before we take a short break, Dr. Dolores Davisio. And while Dolores sets up, and she's on mute, by the way, Dolores, I'm going to introduce you. So Dr. Davisio is a pathologist by training and a molecular and cell biologist who trained at Albert Einstein College of Medicine and Harvard Med School. She's currently holds faculty at Harvard and also has an academic appointment. Her lab is at Cedars-Sinai, which is in Los Angeles, and she is an associate professor. Dr. Davisio is one of the perennial researchers who studies these oncosomes that Peter mentioned and what I mentioned, and these are those particles that are shed by cancer cells. Her team currently studies those at what we call EVs at Cedars-Sinai, and she has, she's really, one of the top researchers, and I really look forward to hearing what you have to say. So Dolores, the floor is yours.
Thanks, Josh, for the nice introduction, and thank you and the other organizers for putting together this panel that I'm very excited to be part of. So as mentioned already by Danny, by Peter, by Josh, there are these extracellular vesicles that are membrane-derived vesicles released by any cell in the body. And I will give you a very brief introduction, mentioning that the importance of these vesicles is that they are released by certain cells, and they can be taken up by other cells that are influenced by these vesicles. And if we imagine a cancer cell in particular, we know that there is a wide range of extracellular vesicles that can be released by this cell, and they can be taken up by all cells in the tumor microenvironment that are affected and reprogrammed in a way that ultimately promotes the cancer progression to metastasis. And I like to use this slide, this schematic, that really depicts the heterogeneity. So not only the cancer cells are heterogeneous, but also the vesicles deriving from these cells are highly heterogeneous. And while most studies historically have focused on very tiny vesicles known as exosomes that originate by a distinct pathway that involves the endocytotic machinery, the formation of multivesicular bodies, and the fusion of these bodies with the plasma membrane for the release of the vesicles. We now know that there are other type of vesicles that originate directly from the plasma membrane of the cells by direct budding, and several of these vesicles have been already reported, and this includes the large oncosomes, which are significantly larger than the exosomes. And we were the first to report the presence of these vesicles, specifically from highly aggressive cancer cells that transition from a mesenchymal type of motility to an amoeboid type of motility that Danny has mentioned at the very beginning. And so we demonstrated that the loss or the silencing of cytoskeletal regulator DIAF3 induces the transition of the cancer cells from the mesenchymal to the amoeboid phenotype and results in the shedding of these very large extracellular vesicles. We found them particularly interesting because when we started looking at patients, and in this case we worked on prostate cancer patients, we noticed by looking both in the tissues and in the plasma of these patients that there was a significant correlation between the presence of these large vesicles and progression to metastatic disease. In this case, we have castration-resistant prostate cancer, which is metastatic. We also used some animal models that basically gave us the same result. When we look at particles larger than 1 micron, which is the case for the large oncosomes, we have a significant correlation with metastasis. And this is not the case if we look at the exosome-like particles, because we know that all cells in the body release these small particles, not only the cancer cells. So a part of my laboratory is interested in trying to use these particles to develop a novel approach to liquid biopsy that Peter has already been talking about in depth. So I'm not gonna go too deep in this talk on the liquid biopsy approach. However, one advantage of these vesicles is that they can be found in the circulation, in the blood of patients with any type of tumor, not just with prostate or breast cancer. And they can be, once isolated with specific procedures, they can be analyzed at the protein, DNA, or RNA level. In fact, one of the advantages of using these vesicles as a source of biomarkers in the liquid biopsy approach is that we can look at different type of molecules that are enclosed in these vesicles, from DNA to RNA, but also proteins and lipids that all seem to play important functions in these vesicles. So we published a few years ago a paper demonstrating that when we compare the abundance of RNA in large oncosomes versus exosomes, we find significantly more abundant RNA in these large vesicles. The profile of the RNA in these vesicles is more similar to the profile of the originating cells versus the profile of the exosomes. We were able to do a whole transcriptome analysis using an Illumina ISIC2000 platform at long time ago, and we were able to demonstrate that there is a significant number of reads of these messenger RNA transcripts in large oncosomes more than in the exosomes. And when we compare the profile of the RNA in exosomes and large oncosomes, we found a very striking similarity. So both vesicles, if they are derived from the cancer cell, contain similar transcripts with some differences that we are now analyzing more deeply. We also saw that the profile of the exosomes and large oncosomes is different from the cells of origin. And I think this is a very important point that is in favor of using these vesicles for the liquid biopsy, because cancer cells can shed material that is not necessarily highly expressed in the cells themselves. So we can use the blood to basically identify altered molecules from the cancer that the cancer cells are spitting out basically during the progression. We also demonstrated by using by doing an in silico analysis using transcriptome data from the ENCODE cell line compendium that has specific analysis of the cytosol, nuclear, and whole cell, that the profile of the of RNA in these large oncosomes is more similar to the profile of the RNA in the cytosol versus the nucleus, which is important because it can help us understand the origin of these RNA in the vesicles. We also found that there is a significant enrichment in the vesicles in comparison to the cells of origin for transcripts that encode for transcription factors that are important in promoting cell division and inducing a transition from the G1 to the S phase of the cell cycle. And we also found when we looked at the patient plasma. So we isolated these vesicles also from the patients of from the plasma of 10 patients with breast cancer. We found that there is these transcription factors are significantly upregulated in the vesicles from the patient plasma in comparison to the controls. And finally, again, we were very lucky to extract these vesicles from patients with breast cancer. We found that when we looked at all the altered transcripts in comparison to the controls in patients, we found, and we compared them to known transcripts that are upregulated or downregulated in tissues from patients with breast cancer. We found a significant correlation between the blood and the tissue, suggesting that we can use these small vesicles as a readout and as a non-invasive biopsy that can be potentially used also for monitoring. So I just want to reconnect very rapidly to Dr. Welch talk, where she nicely showed all the data on the PDX and using the PDX as a monitor of the response to chemotherapeutic agents. Obviously, the extracellular vesicles could also be investigated in those mice or in other models to basically have an idea in real time and without any invasive biopsy of the response of the tumor. I also want to mention another study that we published a couple of years ago and some unpublished data following that up where we demonstrated that large oncosomes are the vesicles in the plasma that contain most of the DNA. So they are suitable for genomic analysis. And we have now ongoing experiments where we are comparing the results of these liquid biopsy using large oncosomes versus circulating DNA. And what we are finding is that if we use very deep sequencing of all exome sequencing in the blood of patients, we can identify deletions and amplifications or copy number variations that are typical of the tumor, in this case prostate cancer, and can be easily identified in patient plasma in real time. We also can simply look at the abundance of these particles by using tunable resistive pulse sensing, which is a new technology that allows to quantify and measure the size of these vesicles. And when we look at the vesicles larger than 1 micron, as is the case of the large oncosomes, we can see that the abundance of these particles in patients, and we have here 4 patients with lung cancer, is significantly higher than in the control individuals. So we can easily, I think we are now ready to move to the clinics, and my lab is trying to develop clinical tools that are easily to implement in a CLIA lab. So regarding the function of these vesicles, which I think is also very very interesting, what we have shown a few years ago is that large oncosomes, but not exosomes, isolated from plasma of patients with metastases, are enriched with a very important and active kinase in cancer, which is phospho-AKT. So we can easily identify this protein, this functional protein in the vesicles. When we use large oncosomes to condition cells in the tumor microenvironment, as Josh was showing with all of our tools of cell culturing and transwell assays, etc., we show that the large oncosomes, but not the exosomes, induce activation of one of the most important oncogenes, which is MYC. And that this is dependent on the presence of the kinase in the oncosomes. In fact, when we inhibit the kinase, we see this phenomenon disappear. We also show that large oncosomes can induce activation, expression of alpha-smooth muscle actin, which is a marker of reactive stroma, which is an indication of progressive cancer. And this is inhibited by inhibition of MYC. These vesicles can also induce angiogenesis, as shown by a specific in vitro assay, and we can also inhibit this phenomenon by inhibiting MYC in the stroma. And finally, when we also did a mouse experiment in which we injected the cancer cells with the stromal cells together, and the pre-treatment of the stromal cells with large oncosomes significantly increased tumor growth. And when we inhibited MYC, there was a complete suppression of these phenomenon. We are now moving forward to a model that is more representative of metastasis, because we know that both in prostate and breast cancer, metastasis can occur several years, if not decades, after the primary tumor. So we're now using bone marrow mesenchymal stem cells, and these are primary cells isolated directly from human individuals. And what so after characterizing our vesicles using rigorous methodologies, we have basically shown that if we treat these cells with exosomes and large oncosomes, we can see a strong upregulation of mRNAs codifying for chemokines and cytokines that are very important in tumor metastasis. And this is a dose-dependent effect that is significantly more powerful when we treat the bone cells with oncosomes in comparison with exosomes. We have now performed large-scale analysis in this bone marrow mesenchymal stem cells treated with large oncosome and exosomes, and we show that the number of genes that is influenced by large oncosomes is significantly higher than those influenced by exosomes. And we have also shown that so this is also evident here in these heat maps that show all the genes that are influenced by exosomes here and large oncosomes here. As you can see, the signal is much stronger if you look at the intensity of the red and the blue in large oncosomes treated cells than exosomes treated cells. And if we look at the top 50 genes that are upregulated and the lowest 25 genes that are downregulated by these vesicles, we find that the most important alterations have to do with the most important pathways that are altered by these vesicles have to do with neutrophil migration, with cell chemotaxis, and with an inflammatory response. So as Danny had mentioned in the introduction, the tumor microenvironment is very important in the response to the tumor. And we are really understanding more and more how different type of immune cells can respond differently to the tumors in ways that probably initially try to block the tumor, but then eventually result in promoting tumor progression as well. So we have done experiments where we show that both exosomes and large oncosomes can induce, when they are used to treat the bone marrow mesenchymal stem cells, can induce migration of neutrophils and cancer cells, suggesting that they might be recruiting these cells to the bone to basically generate what is known as the pre-metastatic niche. So in conclusion, what I've shown you today is that extracellular vesicles are not just exosomes, but they can come in different sizes. They have distinct cargo molecules in them, and they can play distinct functions. The large oncosomes are cancer specific, and they contain cancer-derived molecules that can be interrogated in patient blood. Large oncosomes can reprogram the cells in the tumor microenvironment, inducing an inflammatory response and other modifications that are pro-metastatic. Of course, we need additional investigation to really understand how these distinct pathways can be altered by distinct vesicles, because one of the ultimate goals of our research is also to try to understand how these large oncosomes are formed and how we can block them, because obviously by just blocking exosomes, we're not going to block all the intercellular communication that occurs between cancer cells and the tumor microenvironment. And I want to thank first of all the students and postdocs in the lab who performed the actual work, and several collaborators that have been very helpful in these past 10 years, the funding agency, and obviously the patients for sharing their specimens. And thank you for your attention.
Thank you, Dolores. That was great and fascinating. So now we're going to move. We're running a little bit over, but that's okay. We're going to go through the questions that we've received in chat. And I know there's several feeds, so I'm just going to read the questions and I'll prompt the researchers to answer them. So our first couple questions are for Alana Welms. So one of the questions is, will 4C, the trial, only enroll patients who have experienced their first metastatic recurrence from early stage or de novo disease, or will it include patients who have experienced failure of metastatic treatment regimen?
Yeah, so that's a little bit complicated question. It depends on, so 4C is a very small study, only 15 patients. It's a feasibility study to make sure we can make the organoids from the metastatic biopsies, get it and find out if we can get drug recommendations. And we're actually interrogating the physicians. So we're doing informed consent of the physicians so that we can ask them to take surveys to figure out whether or not the information we provide actually influences their treatment decisions. So because it's so small, we're really focusing it on first metastatic recurrence in triple negative breast cancer, or progression of ER positive disease after all hormone therapy combinations are exhausted. So it's a needed window, but we're trying to keep it kind of uniform so that with a small data set, we can ask about feasibility.
Got it. Okay, great. I guess the next question is, I mean, it's more around, CTC and biology, but Dolores, Peter, Alana, either one of you chime in, but we can start with Peter first. So one of the questions was, for those whose tumor markers are unreliable, would it be likely that CTCs may not be present in blood samples? And is there a correlation between CTC availability and tumor markers?
I'm going to start answering this question in a research use only context. It's really important now to differentiate between what we call clinical care and research use only. And Dolores briefly mentioned as well that, you know, she is readying her work to be deployed in a CLIA lab, which would mean that it could actually be used, start being used for decision making, which again is what we have done in prostate. So it's really important to separate out what we do as research findings versus what could be used in the clinical setting, because this question is a very specific question in that regard. Tumor markers are always difficult. Biomarkers are always difficult because they are so much dependent on the disease and the therapeutic pressure, and us understanding how they go up and down is really the key challenge that we have to overcome, which is exactly why in this third generation approach, the attempt is really to look at all possible biomarkers of the tumor. So not just one, right? So this is where you see people like Dolores and ourselves actually collaborating, right? Because we want to make sure that we look at oncosomes as well as CTCs, as well as circulating tumor DNA, as well as, honestly, all of the traditional biomarkers, because there will be cases where one of them will be giving us a very telling story, whereas the other one isn't. So there's always advantages and disadvantages when you think of these biomarkers. If they're all available, then, you know, then you pick whatever is best to make the decision. But oftentimes, some of them might not be available for a particular reason in that particular setting for the patient. So I know that this is not a satisfying answer because it's not a yes/no answer, but that really reflects of why biomarker development, tumor marker development is just as complicated as drug development, and has to be taken with the same rigor in terms of clinical trials as we would do in drug development as well, because of course we want to support very direct decision making for the patients.
Thank you. So, so my, I'm going to ask a couple, two more questions. Are there any open / enrolling liquid biopsy trials for metastatic patients who have no evidence of disease? I think, Peter or Alana, I don't believe it, that there are any.
So no, and part of this question that is being raised is actually a broad question as well, right? Because and we talk about this, every week. Actually, I had a conversation about this yesterday. Hey, Peter, if resources would not be an issue, how would you support breast cancer patients? What's the trial that you would actually run? And I said, oh, straightforward. Anybody who has ever gotten an Oncotype DX done, offer them to participate in a overtime trial, because that is what we need to study. Right. So the good news for that patient with no evidence of disease is, of course, that, well, no evidence of disease means that there is nothing clinically visible. So when we go subclinical, meaning something that we might be able to see in the blood, we would love to follow a group of patients like that, right, to prove whether or not we can identify recurrence early, and if we intervene early, whether we can improve that patient's life. And in order to do that, we have to do this as these larger scale trials, which are so desperately needed and really need to be open for patients around the country. And which is what I said in the very beginning when I said in the post-outbreak world, one thing that I think that opened our eyes to is that remote patient enrollment, remote patient consenting, it's all entirely doable. So I really hope that as a scientific community together, and when I say scientific community, I include all patients in this because you're a huge driver, that we can really move forward with this.
Okay, great. And I think, I agree. I think, when patients, we say no evidence of disease, it doesn't mean that it's not there. It's just we don't have those thresholds, right, to look at the cells, on a, we do have the threshold, but it's not so finite and refined right now to see it, on that space. So, and the way I always the analogy I always use is TV resolutions, right? So the higher resolution we get, the more we could see, and the more we can find, that needle in the haystack. The last questions, and I'll let go because we're going to be short on, we're running over onto the other panel, is, there's this question that's been asked is there a case to biopsy metastasis when possible? and if it can be different from the primary tumor. And yes, that is the case. We need to have more, as far as Alana's trial goes, from the point of view of Peter and Dolores's research, we need to get the metastatic tissue. We need to find out, take those out, biopsy them, so that we can compare. Remember what initially Danny and I talked about. There is a difference of the primary versus metastasis, and there are the different marbles that go hand in hand, and we need to figure out how is that tumor evolving. And so in most cases, it's more important to know the metastatic tissue landscape than it is the primary, because tumor cells have evolved and now there's treatment resistant going. So that is the goal, and I hope that we've conveyed that to you in this first session for this morning/afternoon. And so with that, I really want to thank Danny, Alana, Peter, and Dolores, really for fantastic talks and updating us on their research and where the field of metastatic breast cancer research goes. I'm sure all of the patients and participants out there are really thankful for your participation. And so with that, I want to thank you, and I hope you guys enjoy your afternoon, or Alana, your late afternoon in Utah. And
I'm gonna listen to the rest of the talks.
Okay, so if you're staying, great.
Josh, thank you, Josh. I saw the lineup, listening to Saul and Pat and everybody who was lined up in the afternoon. I'm definitely staying on.
Yeah. Okay, good. So, and so, I know we were supposed to have a 10-minute break, but like everything, we're all used to it. Conferences go over. We're just gonna, I guess we're just gonna go straight into our next session. And I'm gonna introduce, our second panel, of speakers. With that, our first speaker in the second session, which I know there was not a research pause for it, is Dr. Cyrus Gajar. Hi, Cyrus, how are you? I think you're, hopefully, let him get on. I see Carrie.
Hi.
Oh,
How are you?
How are you? Great to see you.
Good to see you. Nice to see you too.
Good.
Thanks
good.
for having me.
Of course. I think you have the access to share your screen while you put your slides up. I'm going to introduce you. So Cyrus Gajar, is an associate professor of human biology and public health sciences division of the Fred Hutch in Seattle, Washington. He's the lab director for the laboratory to study metastatic microenvironments called LSM2. Very cool. Cyrus is, he's interested in studying how distant tissue microenvironments that we've been talking about, and then dormant cells, which are called disseminated cells, basically escape and elude, therapy, and they set up that environment that is necessary. He is one, again, of the leading researchers, and I'm always very fascinated and look forward to his talks. So Cyrus, with that, the Zoom floor is all yours.
Well, thank you, Josh, and thank you for the opportunity to participate. It's an honor and a privilege, and I look forward to the discussion and seeing everybody else's talks as well and the interaction afterwards. So what I want to talk to everybody about today is targeting dormant disseminated tumor cells for the purposes of preventing breast cancer metastasis. I want to start with an acknowledgement of my lab. This is as 2020 as a picture gets. We have a socially distanced picnic, and you can tell by the red sky there were fires in the Seattle area, a lot of smoke, so we were affected by all of that. But we have a wonderful lab, and we're back to full speed and working as hard as we can to make progress, because as important as it is to stay healthy and safe, it's just as important to advance our cancer research as well. So the metastatic cascade, I'm sure you've heard a lot about it in the first session, is typically depicted as this linear process where the primary tumor grows, it becomes invasive, and then eventually it enters the vasculature and then exits at secondary sites like the liver, the bone marrow, brain, lung. And what we're often taught in the textbooks is that if tumor cells can't survive at this secondary site, then they're able to adapt and proliferate to form metastases. This linearity of the metastatic cascade and the depiction of it in this fashion has really led to approaches where we try to target invasion, because it was thought if we can block this step, it's the most genetically aberrant or mutated cells by the time a tumor is detected. If we can block them from invading, then we'll be able to prevent metastasis. And this has led to maybe a disproportionate amount of focus on trying to block processes that lead to invasion, MMP inhibitors, for instance. And by and large, these haven't really worked to prevent metastases. So we need more of a focus on what's going on at the secondary site. We need better therapies against metastases, but we also, I would argue, have a window of opportunity to do something about metastases before they arise. Because in breast cancer, we have these characteristic relapse peaks 18 months after treatment, 5 years after treatment, and then continued relapse after 5 years of treatment. And an increasingly large proportion of patients are relapsing later and later. So what do I mean by an increasingly large proportion? This is a study published in the New England Journal of Medicine at the end of 2017. And this is a huge retrospective analysis of about 70,000 women who were treated with 5 years of endocrine therapy. So these are estrogen receptor positive breast cancers, and these women were treated with 5 years of endocrine therapy. And what you can see when you look at recurrence as a function of lymph node status, no involved lymph nodes in this kind of orange, yellow color. I don't know what you would call that, mustard, perhaps. Tangerine, I don't know. You have some nodes involved in blue, and then extensive nodal involvement in red. And what you'll note is that you have recurrence 5 years, but if you look at recurrence going through year 20, the majority of recurrences are taking place after year 5. And what this is suggesting is that therapy is not curing these patients per se, but is putting off relapse into the future. And so I'd argue that we have this window of opportunity between the time the tumor is detected and the time that these metastases show up, maybe years or even decades later, to do something to target the seeds of metastases. And I'd also point out that currently we don't have a single therapy that's really aimed at selectively targeting the seeds of metastases. And this is, to me, a huge challenge and a huge opportunity to be able to really impact patients and prevent them from ultimately presenting with metastases. So what are we trying to target? From the time a breast tumor cell shows up in an organ like the lung to the time it becomes clinically detectable, what is it doing? Well, it could be doing a couple things. One is that it can exist as a dormant mass, so proliferation and death being balanced at roughly the same rate. The other thing that can happen is that it can exist as a cell, a single cell or a very small cluster of quiescent cells that have exited in the cell cycle and remain quiescent. And you can imagine, this isn't semantics. If you want to target a mass of maybe tens of millions of cells that are growing and dying at the same rate versus targeting a single cell that's quiescent, the biology that you're trying to target is going to be very different. So I think it's a very relevant question to ask what is actually present in an organ at any given time. And what we know the most about is bone marrow, because it's accessible. We can take a needle and put it into somebody's hip and actually suck out their bone marrow and stain for cytokeratin to identify cells of epithelial origin, cells that have to have come from the breast epithelia in a breast cancer patient and reside in the bone marrow. And by and large, this is what we find: single or small clusters of these red cytokeratin positive cells that are persisting in the marrow. And we know if they're there that they portend poorer metastasis-free survival, meaning these women are more likely to get metastases. This hasn't been studied in men with breast cancer, but I would venture that maybe we would find out the same thing. So is this what we want to target? Do we want to target these dormant cells? Are they really the cells that give rise to metastases later on? This is an assumption, and I'm very upfront about saying this is an assumption. And when I started my lab, this is absolutely an assumption that we wanted to test. And the way we went about that was with intravital imaging. So what this allows us to do is actually remove a piece of the skull and actually put a window on a mouse's brain. The mouse is perfectly fine afterwards. As you can see, if you did this to me, I don't think I would be perfectly fine with it. But mice just soldier on. They stay alive for weeks and weeks. They would stay alive indefinitely if it wasn't, for the fact that we're about to inject them with tumor cells. And what we can do, though, is inject them with fluorescent tumor cells. And now we can look inside of the brain, and you can see the kind of pictures we're able to take now inside of the brain to visualize cells arriving into the brain, these green cells in the red blood vessels, getting out of the vessels and actually setting up shop around the vessels. So what we wanted to do was take a line that could not form brain metastases and a line that could form brain metastases. And we wanted to make a comparison of where exactly the metastatic process fails in the cells that cannot form metastases versus those that do. And so I'm going to show you data for one line, and we've done this with 2 different lines, and we get the extremely similar results. So you can see that in one line that is not able to form brain metastases, these cells persist in the brain, and you can see single cells that are just aligned on the blood vessels and remain in the brain for over 6 weeks in this case. And then we have lines that will form, these are the same line, in this case is MDA and B231, and then a brain metastatic MDA and B231 that we actually got from Patricia Stieg, who I see kind of underneath me to my left. Thank you, Patricia. Thank you, Pat, for this line. And so they form brain metastases. And now we've done this, as I said, with 2 different lines, T42 and MDA231. And then what do we see? What we see is that the latent lines in green and the brain tropic lines in blue, they don't differ in their ability to enter the brain. They enter the brain at roughly the same rate. They don't differ in their ability to survive in the brain, which is often called a very hostile site. They basically die at the same rate upon entry into the brain. Where they differ is in their ability to escape this phase of dormancy, progress towards micrometastases, and sustain those micrometastases. And what that argues is that this is a rate-limiting step, or maybe perhaps the rate-limiting step of metastatic progression, and the one that we want to target. And if we could target it, perhaps then we could prevent metastases. We could catch cells in the state and kill them. So what are the hallmarks of these cells? What can we understand about them and what can we target? Well, 1, we know that they survive for very long periods of time. And 2, and despite being from the breast and existing at this distant site. And 2, we know that they're growth arrested. And if we care about these cells, they must be reversibly growth arrested. They have to be able to wake up again. So what can we learn about that? Well, I note that one thing I want to point out is that each and every cell that gets into the brain, this isn't even a 999 out of 1,000 thing. We've actually tracked over 1,000 cells this way. Every single cell we've tracked actually sets up shop, not near the vasculature, but on the vasculature. And that's where they persist. And ultimately, no matter what they do, they have to be in contact with the vasculature. We don't note cells even ever leaving this niche. And this really is something that holds up regardless of where we look, whether it's the brain, liver, lymph node, bone marrow, or lung. And so what this has allowed us to do is actually try to understand these niches. We model them in culture. We create human vasculature in a dish, and then we study the interactions between the vasculature and the tumor cells. And we're able to recapitulate this phenomenon of dormancy or breast tumor quiescence in the dish. So a couple things that we've been doing is, number 1, trying to understand what enables these cells to survive. We've run high throughput screens, meaning we are able to screen over 5,000 drugs in these microenvironments and look for drugs that selectively target dormant cells. And we've made a lot of progress in that area. It's actually very serendipitously making sense and harmonizing with another totally unrelated project we have in the lab. And we think that we've identified unique hallmarks of dormant cells that would allow us to kill them, perhaps without chemotherapy. We've also used these niches to try to understand what's actually putting these cells to sleep. If we could understand that, maybe we can actually just enforce these cues and keep these cells asleep indefinitely. The very interesting thing is that the mechanisms that seem to induce dormancy in lung and bone marrow differ from those in the lymph node, something that you would expect given that lymph node metastases are so common. What regulates those cells and keeps them dormant probably should differ from other organs. And they're totally different than from what we've observed in the brain. In the brain, the burden actually seems to shift to perivascular cells called astrocytes, and they're inducing dormancy through a protein and through receptors that don't seem to matter at other sites. But a third hallmark of these cells is that they're chemoresistant. And so in the time that remains, what I want to do is tell you what we've tried to do to understand the fact that these cells that are asleep are resisting chemotherapy. The assumption has and really continues to be that cells that are not dividing are not sensitive to chemotherapy. So cells that are dormant don't respond to chemotherapy because they're dormant, not because of the environment that they occupy. We wanted to challenge this assumption. And the way that we challenged it was trying to understand if we treated mice with dose-dense chemotherapy the same way that a patient with invasive breast cancer would be treated with doxorubicin and cyclophosphamide, where would the cells that survive reside? So we looked in the bone marrow, we found disseminated tumor cells, and we mapped their distance to the vasculature and to other cell types. And the bottom line is what we found, that with chemotherapy, this actually selected for cells that were even closer to the vessel than they were to begin with. And you can see here, all these red dots collapse to the x-axis, and chemotherapy is really forcing these cells or selecting for the cells that are already sitting on the vasculature. So we wanted to study what it is on the surface of the vasculature that could be protecting these cells from chemotherapy. And we did a number of things, and we narrowed it down to some proteins that signal through molecules called integrins, specifically an integrin called integrin beta 1 and another integrin called integrin alpha V beta 3. And so what we did then was think about a way that we could actually trial this in a preclinical setting. We took a model of luminal B estrogen receptor positive breast cancer and delivered these cells into overectomized mice. So what we're trying to do is model estrogen suppression and progression under the context of estrogen suppression. And then we treated mice with this the adriamycin cyclophosphamide regimen, and then and either preceded that chemotherapy with just a generic antibody or antibodies targeting integrin beta 1 or integrin beta 1 and integrin alpha V beta 3. And what we did first was assess the burden of disseminated tumor cells in the marrow. And then the second thing we did was actually see if this would help metastasis-free survival. So this is the bone of a mouse treated with chemotherapy preceded by generic antibody. This is the femur. You can see all of these green cells lit up in the femur and how much burden there is throughout the femur. This is the femur of a mouse treated with integrin inhibition in combination with chemotherapy. And now you can see that you see some green cells kind of scattered throughout, but they're really few and far between. So we've decreased the burden of disseminated cells in the marrow of these mice by 94%. And then the question is this actually going to lead to better metastasis-free survival? And the answer is that it does. And you can compare that the mice treated just with chemotherapy alone, over 70% of them recur with bone metastases and some with adrenal metastases. And that's in contrast to those where we preceded chemotherapy with this integrin beta 1 inhibitor, where only about 22% of the mice succumb to bone metastases or actually experience bone metastases down the road. And so the question is, okay, well, now we're kind of augmenting chemotherapy with an inhibitor to a very broadly expressed molecule. Does that have an adverse effect? And it doesn't in terms of body weight, in terms of vascular density, and in terms of bone marrow cellularity, where we see that, yes, chemotherapy has an effect on some of these metrics, but adding integrin beta 1 inhibition in the context of chemotherapy does not change that or enhance any of the chemotherapy associated toxicities. So I will end there and say that we can conclude that the perivascular niche is protecting tumor cells from chemotherapy, and it is not because of their proliferation state, but because of the environment created in the vascular niche, and specifically through molecules deposited around the vasculature that are signaling through integrins on disseminated tumor cells, protecting them from chemotherapy. And when you interrupt these cues, you can now sensitize cells to chemotherapy. And the key is that you do this without waking them up. I didn't show you guys that, but we are sensitizing them to chemotherapy. And by inhibiting integrins, we are not actually waking these cells up or causing them to cycle. And this seems to occur, at least in mice, without undesired side effects. So the question is, we work with patient advocates quite a bit, and they're very adamant about the fact, I was really impressed with this, and I was very excited to show them the data. And I'm like, well, this is great, but we don't want chemotherapy. And this is why it's wonderful to continue talking to patients, because, I might have been satisfied with this result. Otherwise, now I'm actually not satisfied with this result. How do we remove chemotherapy from the equation? As I've said, we've screened for drugs that specifically kill dormant cells in the absence of chemotherapy. I think we have some very, very promising leads that are potentially targeting new or at least previously unknown biology of these cells. And we're also now trying to discover immunotherapies and how to sensitize these cells to immunotherapies. And we are specifically focusing on CAR T cells for a variety of reasons that maybe I'll get into next time. So thank you for your attention. Thank you for allowing me the opportunity to speak here. And I'll look forward to the panel at the end of this.
Thanks, Cyrus. That was great as always, and very relatable. So just to stay on course, we're going to have our panel again at the end. We have a couple free questions, but while Saul sets up his talk, I'm going to introduce him. So Dr. Saul Priceman is an assistant professor in the Department of Hematology and Hematopoietic Cell Transplantation and Associate Director of Translational Sciences in the T-cell therapy program at City of Hope and the Beckman Research Center. Saul is, a card-carrying immunologist, and his research focus is on CAR T-cell therapy, and he has some of his therapies that are now in clinical trials, but I will let him tell you that part of his talk. And with that, Saul, the floor is yours. You were in presentation mode before.
I was and realized I was muted, and so
that's okay.
Figure out how to get back here. But hello.
Great to have you. Oh, you're on the bottom right. Go to bottom right where this. Go lower.
Sorry about that.
We can always cheerlead in the background, so
yeah.
There's all kinds of stuff.
Please do, Peter, because I'm in trouble here. I'm going to try again. Hold on 1 second. As you can tell, I'm an immunologist and not a computational biologist. Can you see my slides?
I could see your slides, yeah. Well, no, we could see if you have two screens, I think we could see, we don't see your, oh, there you go. Now we see it.
All right, wonderful. I don't know what happened, but thank you, Josh, for the introduction, and thank you to the organizers for the invitation. It's a pleasure to be here. And so I was going to go through and give you a little bit of a background on immunology in general and how we've landed on several modalities or therapeutics to target the immune system. And then get into what we're doing specifically in my lab and as an institution at City of Hope in terms of CAR T-cell therapy. And so when we think about therapeutic modalities, everyone will be familiar with surgery and radiation therapy and chemotherapy, and that includes targeted therapies. And in this audience includes things like Herceptin or tucatinib or other HER2-directed therapies, as well as others. And immunotherapy has really come on the scene in the last decade or so as a really viable therapeutic approach at not targeting the tumor cell directly, but rather evolving the immune system to see and target the cancer cells wherever they may be in the body. And so this has taken on several shapes. I'm going to go through them very briefly, and I'm happy to take questions, and I think these slides may be available at the end. But antibody immunotherapy is probably the oldest type of immunotherapy. And if you think of Herceptin, which is an antibody that targets HER2, an oncogene that's overexpressed in breast cancer, not only does it target the HER2 pathway to target a pathway in the cancer cells that's required for their survival, but also it engages the immune system as well. And that's done by a specific domain or protein segment on the antibody that engages immune cells that allow them to also target the tumor cell and cause cell death. And we've thought about cancer vaccines. This stems from all of our knowledge about vaccines in the field at targeting infectious diseases, and especially what we're doing now in terms of trying to get a vaccine for the treatment of COVID-19, as you all have been following the news as I have. But in terms of cancer vaccines, this started really in prostate cancer, actually, with a drug called Provenge, which took an immune cell, taught it that tumor cells were there and what they were, and then infused those in what's called autologous, meaning we took the immune cells from one patient, we trained those against the tumor cells in that patient, and then we put them back in, and hopefully those immune cells went around and found the cancer cells and killed them. And that was FDA approved, I think, in 2012. And then we generated more targeted therapies, which, as brutally as possible, took an immune cell, took a cancer cell, and forced them to come into close proximity. And these are called bispecific T-cell engagers. And that's shown here, and allows this specific immune cell, we call a killer cell, a T-cell, to see the tumor cell and engage the killing capabilities it has when it comes into contact with a virally infected cell or hopefully a cancer cell. This is just a therapeutic modality, and that has been FDA approved now for the treatment of hematological malignancies or blood cancers, and is being tested actively in solid tumors, including breast cancer. And then checkpoint blockade, which is the idea that a tumor cell should be recognized by an immune cell. It probably is being recognized by the immune cell, but the cancer cell has evolved a way to shut off the immune system's ability to target it. And those are checkpoint pathways. It's the same pathway, and I could go into a whole spiel about this for another hour, but it's the same pathway that allows the resolution or the contraction phase of a normal immune response once you've cleared an infection. You don't want your immune system to continue going and going and going. You want it to shut down. Well, cancer cells is forcing the immune system to shut down. Those are checkpoint pathways. And we've found antibodies, and that's the royal we, that we found antibodies and essentially release the brake on the immune system so they can go around and target those cancer cells. Again, that's very active in the melanoma space, a skin cancer, and has been FDA approved now for multiple disease indications, including most recently, I think, triple negative breast cancer. And so those are very viable approaches that we're looking at. We took a different approach, and this was studies that started in the early 90s and have matured over the last 30 or so years, where we've understood what T cells do when they engage a virally infected cell or a cancer cell. They should target it very specifically, but cancer cells have evolved ways to shut that down. We essentially built out a chimeric antigen receptor, which allows the engineering of a T cell in a way that can force it to see that cancer cell again. And I'm going to give you an example of that in breast cancer. I'm going to skip this video because I think I've talked too much. But to suffice it to say, this is a T cell in green targeting a tumor cell, secreting enzymes that literally pops holes in the membrane of the tumor cell and go on and do that serially. And that's something unique. We call it a living drug. It doesn't need to be infused over and over again. It's infused once, and the immune system grows out, hopefully, to target the entire cancer cell. This is the landscape of where we are with immunotherapies, and you can just see there are hundreds of early to late clinical trials that are testing all different types of therapeutic modalities, including cell therapy, oncolytic viruses, which I'm actually going to give you a little teaser about at the end, and other therapies that are re-engaging the immune system. And it started really with CD19, which is a protein that's expressed on the surface of B cells, another immune cell in your body, that gives rise to a lot of lymphomas and leukemias, which are very devastating hematological malignancies. The field built CD19 CAR T cells that target CD19 on those B cells, those malignant B cells, and get rid of them. And this is just a scan of a patient that was treated at City of Hope with a CD19 directed CAR T cell. This is the pre-treatment scan on the left. I don't know if you see my mouse. And on the right, 28 days later, one infusion of these CAR T cells eradicated disease all around the body of this patient. And we've seen that dozens of times now. And you can see in the table there, it's 100% responsive in these patients, which is a remarkable response. It's actually landed in the field in several FDA approvals for the treatment of heme malignancies in patients with quite staggering response rates. And these are heavily pretreated patients. They may have seen 5 to 10 prior lines of therapies, including bone marrow transplants, et cetera. So they're really on their last straw, and they come in and they have some remarkable therapeutic responses that can be durable in many cases. Our program has done very similar. This is a schematic we published 6 years ago now in the autologous CAR T cell world, where it starts as a leukapheresis, a large blood draw. We engineer those T cells to express the chimeric antigen receptor. We grow them up in dishes over the course of a week or two, and then at some point we administer them back into the patient. We've done this for over 200 patients in the last 4 years, spanning hematological malignancies all the way to solid tumors. And if you look in the middle in BBM, it's breast to brain metastasis, which I'll talk about today. And so there's a huge clinical unmet need. Over the last decade, there have been several HER2-directed therapies for the treatment of these patients. But really, at the moment, tucatinib, which just got approval several months ago, is currently the only approved therapy for the treatment of patients with HER2-positive disease, and as well as brain metastasis. And immunotherapy, CAR T cell therapy in particular, has tremendous promise for the treatment of these patients. And because of that, we developed in our lab a HER2-directed CAR T cell for the treatment of patients with HER2-positive brain and or leptomeningeal metastasis. And this was based on multiple preclinical or mouse data, and I'm showing that here. You can see the colorful blue, red, green spectrum. And that really just highlights the tumor cells growing in the body of a mouse, in particular in the brain. And we treated them with intraventricular delivered HER2 CAR T cells. So these are CAR T cells that we put into a catheter into the ventricle in the brain, which houses the cerebrospinal fluid in the brain, or bathes the brain. And those T cells traffic to the tumor and eradicated disease in 100% of the mice treated. And this was very spectacular because when we did the same thing but gave the CAR T cells intravenously, systemically, at a 10-fold higher dose of cells, they were only able to cure 50% of the animals, suggesting that regional delivery for patients with brain metastatic disease may be the most viable approach. And we went on to show that these CAR T cells actually trafficked from the brain out to the brain to treat bone metastatic disease that was extracranial or not within the brain region. And actually these cells were also provided to us by Patricia Stieg and one of the collaborations we've had. And we went on to show that intraventricular delivered CAR T cells cured all the brain metastases, as well as prolonged survival of the mice greater than intravenous delivery. And because of that and a number of other data, we started a clinical trial about a year ago in treating patients with brain and or leptomeningeal disease by intraventricular delivery of HER2 CAR T cells. This is a trial that's supported by the California Institute for Regenerative Medicine. But the major issue in solid tumors is this immune suppressive microenvironment. And it's a complex system. I'm showing this slide because it is a complex system. I'm gonna shorten the point by saying that cancer cells don't like to be targeted by the immune system. They shut off immune cells, including CAR T cells. And we term this as immunologically warm or cold melanomas, which are responsive to what I talked about before, which is checkpoint blockade, respond pretty well because they're immunologically warm. There are already immune systems in the tumor cell waiting to be released. In the context of prostate, pancreatic, breast cancers, they're immunologically cold. There's not a lot of immune response happening in the tumor. And so they're much more difficult to treat, we've found, particularly with checkpoint blockade, but even with CAR T cells. And just a little teaser, there are oncolytic viruses. These are viruses that very selectively target tumor cells. And it's been used in the field to target many cancer cells because they selectively replicate in these tumor cells. They can cause cell death and immunogenic cell death, which actually spawns an immune response, not only to the virus, but to a tumor. And we've taken advantage of that. They also are vehicles that we can deliver genes of interest to the tumor specifically. And I'm going to tell you a bit about that. And TVEC is an FDA-approved oncolytic virus that's treating metastatic melanoma. And though it's promising, there are challenges with single targeting therapeutic agents, even including oncolytic viruses and CAR T cells. And we took this idea and we said, well, if CD19 is such a beautiful target for hematological malignancies and causes durable curative responses in patients with blood cancers, why can't we take a CD19 CAR and redirect it to a solid tumor? How can we do that? Well, we took an oncolytic virus, we forced it to deliver CD19 and infected tumor cells. Those tumor cells then express CD19 on the surface, and we could target them with a CD19 CAR T cell, making a tumor agnostic but very viable combination approach using oncolytic viruses and CAR T cells. We just recently published this work, and we actually showed that it worked very beautifully in mouse models. And this is just a beautiful picture, nothing like what Cyrus showed in the prior slide with vasculature, which was so beautiful to see. But this was just showing that these viruses can infect triple-negative breast cancer cells here and pop CD19 onto the surface. You can see that in purple. And those viruses did that very well, so well that they infected cells 100% of the time, presented CD19 100% of the time, but were only capable of killing 30% or so at specific time points. And we took advantage of that completely in showing that the combination, and this is a triple-negative breast cancer model, which doesn't have HER2 on the surface that we can target with a HER2 CAR, very effectively was targeted by the combination of the oncolytic virus presenting CD19 and then CD19 CAR T cells. And this did more. It actually robustly increased immunity in the tumors and immune infiltration. And we're showing that here, and I'm going to skip on probably over already. But what was most impressive about this is when we cured the animals of their tumors, we were able to re-inject new tumor cells that never saw the virus, never had CD19 on the surface, and they were blocked from growing, suggesting that not only we cured the animal of the primary tumor, but we induced what's called protective anti-tumor immunity. And this is long-term immunity against tumor challenge. So if we can think about recurrences following the curative response, this approach may be a viable way to actually durably cure patients. And we're looking at that very closely, and we'll translate to trial as well, because City of Hope has expertise, obviously, in CAR T-cell therapies and CD19 CAR T-cell therapies, but also oncolytic viruses. So as a conclusion slide, we'll learn lessons from our phase 1 trials. I'm just talking today about breast to brain metastasis, but our lab has ongoing trials in prostate cancer, a new trial to be started in the next couple of months in ovarian cancer, as well as pancreatic cancer. And we're addressing the major challenges that we see, including tumor antigen heterogeneity, which Dolores and Peter talked in terms of looking at liquid biopsies. And actually, I didn't mention, but we have a very beautiful collaboration with Peter Kuhn as part of our brain metastatic trial, which we're very excited about. And he's looking at circulating tumor cells in the cerebrospinal fluid in the brain of these patients. And we hope to learn a lot from that. And then I'll end. This is just a picture of my laboratory. Thank God I'm not wearing the same shirt today. And then this is my lab, our program, the T-Cell Therapeutics Research Laboratories program, many collaborators of which Peter Kuhn and others are listed here, and the funding sources that have supported all of this work. So thank you very much.
Thanks a lot, Saul. Great data, great work. I mean, it's a great example of taking something from the lab bench side and putting it into the clinic, something that we all strive for. So I'm going to move on to the next speaker, which is Dr. Pat Stieg, which she really does not need any introductions, but I'm going to try my best to give, a very summary of, of the work that she has done. And honestly, Pat is, when I do brain metastasis research, she is one of, the seminal people, the leaders of the field, and she is, I would say, as they say, a shero to all of us in many, many ways. So Dr. Pat Stieg is the Deputy Chief of the Women's Malignancy Branch and the Senior Investigator at the NIH and NCI specifically. She was really, and Danny mentioned this earlier, she was one of the first to discover the metastasis suppressor gene, specifically called NME. Her work has been seminal in brain metastasis, specifically with breast cancer. And she is one of those scientists that has, like Saul and many others like us on here, has really taken her work from the bench side and made, and pushed it towards clinical trials and has affected patients. So Pat, really a privilege to introduce you. I think you might still be on mute. No, you're not, but the floor is all yours.
Tell me you can hear me.
Nope, you can hear. Yep, you're good. Go for it.
Okay. Hi, everybody. Thank you so much, Josh. It's been wonderful listening to everybody. I'm going to start a talk about brain metastasis, and my colleague, Carrie Anders, is going to follow with more of a clinical presentation. So I'm going to tell you where we are in the lab, and Carrie can follow up with where we are in the clinic. And you've heard about brain mets from a number of speakers, and of course, Josh works in it. Historically, brain mets were very rare. They occurred very late. Patients were often in hospice. They were just treated with some steroids for edema. And it's now almost 40% of those patients who have metastatic tumors that are either HER2 positive or triple negative. That's an astounding rate. And the clinical course varies. For the HER2 positive patients, brain is many times the first site of relapse after the initial metastatic therapy. And what that means is that systemic sites are either stable or responding. The patient's doing well, she has a life, and this is a very unwelcome thing. In triple negative disease, brain mets often occur in the context of uncontrolled systemic disease. And so that's a bit different. Why we all dread them is they pose severe neurocognitive and physical consequences. particularly both from the lesion and the treatments. And the treatments include surgery to actually take the lesion out, 2 kinds of radiation therapy. There's something called stereotactic, and what that is radiation just to the lesion. And then there's whole brain, and that is the entire brain in usually 10 fractions. It was originally used if there were, quote, multiple brain metastases, which was like 3. We're now up to 10, or, you know, depending on your oncologist opinion, that we're treating with stereotactic before we go to whole brain. Why? In a percentage of patients, there is a long-term irretrievable cognitive loss. And it's just awful. Recently in the HER2 space, there are a number of chemotherapy combinations, and I will let Carrie take you through them. Something to remember, once a brain met forms, the patient's at high risk to develop more brain mets. And the other thing I want you to remember is that drugs that have activity, they're FDA approved in the metastatic setting, are often inactive in brain mets, and we want to know why. So I'm going to address 3 different topics in 20 minutes, which is probably impossible, from the lab, where one is still blue sky. We hope to translate it. One is in the clinic, and one I hope will go to the clinic if it keeps looking good. But what we do is, you've heard the only way to really know metastasis is to inject tumor cells into a mouse and ask, will they spread? And we have either optimized or made on our own any number of cell lines. I think there's 7 shown here that are what I call brain tropic. If we put them in a heart ventricle, they go all throughout the body, and they definitely grow, make brain metastases. And the first thing you can see is they all look different. And I think that's a good thing. I think they explore some of the heterogeneity that is the human condition. And they're all different kinds and subtypes of breast cancer. And so we try to use 2 or 3 or more of these at any time for any study, and then we start believing it. I won't go into these details, but when we made our first one, we compared it in multiple respects to craniotomy specimens, surgically removed brain mets, and it looked pretty good. So it gives you a little confidence of what you're doing. And so this has been the backbone of much of our research. So the first question I want to talk to you about is, why are so many therapeutics active systemically, but not in the brain? And there's going to be multiple answers to this question. Priscilla Brastianos, who's up at Harvard, has shown that there are distinct mutations in brain metastases compared to the patient's own primary tumor. So something else is going on, and that can not only affect the metastasis pattern, but the response to drugs. What I want to show you is heterogeneous and low overall drug levels in the brain metastases. Now, I broke this experiment down into a few slides so that we can all follow along. What we did is we took one of those model systems, we labeled it. It's like a lightning bug, it glows green. We injected it into mice, and we let the brain metastases grow. Once they had grown, we injected the mice with 2 different things. One is just a fluorescent protein. It's like a red lightning bug of 3,000 molecular weight. So this is well above the size of many drugs, and we can argue about antibodies. And we also injected the mice with radioactive paclitaxel. So this is a drug commonly used in breast cancer treatment. We let the drug circulate through the mice, and then under deep anesthesia, we perfuse them. What that means is we push saline into them and get rid of all that drug and all that fluorescent protein in the circulatory system. So the only drug that's left is what has moved into the brain or the brain metastasis. So then we section that brain and see where the mets are and how much drug is in them. Okay, so here is a section of a mouse brain. And remember, I told you green lightning bug was the color of the brain mets. And so this brain is riddled with brain metastases. Okay, now if we don't even take it off the microscope, just switch to the red channel. And now we're looking at the 3,000 molecular weight red protein. Did it get into the metastases? What I want you to see, where is my pointer, is that this big red metastasis over here on the right corresponds to this guy, is highly permeable to that Texas red dextran. This area here, almost no permeability to the drug, and it's a metastasis. So you have everything from high to low drug levels in these metastases in the one animal brain. And if you then go to your radioactive imager for that same section, you see the same trend. You see what lit up with Texas red dextran is very high for uptake of paclitaxel. What was generally negative is negative, okay? So what I think we've said is that the entry of drugs or molecules into brain metastases is heterogeneous. I'm not going to put you through the quantitation, but what we found is that only 10% of the metastases that had the highest permeability, the reddest tumors or the most white on the radioactive scan, showed a response to the drug. In other words, it isn't enough for some drug to get in. A lot of drugs got to get in. You don't need some permeability, you need high permeability. And then I want to ask you, because I know you're going to have an opinion on this, is do we want the whole brain to become highly permeable? I don't think we do. I think this is a recipe for adverse events. You don't want paclitaxel running around your neurons. Okay. Or do we want to selectively increase the permeability to brain metastases? And that's what we're trying to do. People can say, does this occur in humans? This is a pre-surgical study where breast cancer patients coming in for medically indicated craniotomies were given an upfront dose of capecitabine in pre-op, and we looked at the level in their tumor, just like we did in the mice. But here it's a bar graph. If you go to 5-FU, this is the metabolite that can go kill a cancer cell, and each color is a different patient's tumor. And here is the same heterogeneity of drug levels that I showed you in the mice. We did that. We've done the mouse studies for 5 different drugs, same conclusion. We did this for 2 different drugs. So why is it that the brain is so crazy? And it's called the blood-brain barrier. And these are the capillaries that are in your brain. The brain has the most capillaries of any organ in the body. And they don't look like a normal capillary has these pink endothelial cells around it to enclose the blood. And they may be all together or there may be gaps between them. Not so in the brain. The endothelial cells are connected by continuous tight junctions, and they express these pumps. Like if something gets in, they pump it right back out. They are surrounded by pericytes, 2 basement membranes in the feet of astrocytes. Every single one of them provides barrier function such that your brain has a homeostatic environment so like neurons can work. And it's when a tumor forms, Cyrus just showed us, they sit on the outside of these capillaries. But what happens to this structure, this blood-brain barrier, which I'm now calling a blood-tumor barrier? The answer is we don't have a clue. And it's more complicated than that. So here's my blood-brain barrier under the normal brain. And you see them surrounded. These are microglia. These are like innate immune cells that monitor, okay, does everything look good? And they'll just eat anything bad. And neurons, which is what the brain needs to do. Besides the blood tumor barrier, when the metastatic brain, and you see the tumor cells here on the outside of it, you get what's either called a neuroinflammatory response or a glial scar or astrogliosis. They've got lots of names for this. But what it is that these microglia get huge and angry, and they try to wall off whatever this is that's growing in the brain, as well as the feet of these astrocytes. They get angry too, and also try to wall it off. And this is probably an evolution, a response to, say, traumatic brain injury. I don't think the brain developed this for brain metastases. You see the same thing in traumatic brain injury. They try and wall it off, and some immune cells come in. We don't have a good picture of that. Okay.
Sorry to interrupt you. I don't think your slides are moving forward. We're still stuck on the brain metastasis permeability slide.
You're kidding me.
Yeah.
Okay, let me stop a minute. I am going to.
Why don't you restart your PowerPoint again and just go to those slides?
Okay. Are we having fun yet?
But I have to tell you, you were so clear that it was seamless. So don't worry.
Give me a break. How do I escape? Let me do one other thing very quickly. And that is see if, okay, then let's go back to this. Hello.
It's okay.
Oh yeah, it's just absolutely fine and dandy. Why can't I not pull this Zoom up? God, I love technology. This is where I need a young person.
Or a joke.
Or
you know, you guys have my slides.
I do. Let me see. Let me see if I can pull.
There's Zoom.
There you go. Let me see if I can pull.
Okay, let me see share screen. And wait a minute, I need.
Okay, why don't you go ahead and share your screen again?
I'm so sorry.
No, it's okay.
If you guys have it faster, do it.
Let me see.
Oh, this isn't going to work because I took off my my VPN.
It's okay. Hold on.
Oh my heavens. Okay, you guys are going to have to put it up.
I don't have. It's okay. I don't have. Darren, do we have her slides?
Yeah, you do. I sent it.
Yeah. Darren, if you have it, can you post it or can you pull it up?
He texted, he has it.
Yeah, no, I know he has it. Let me see. Pat, okay, so Darren is going to pull up your slides. Yeah, he's going to.
Does Carrie want to go and we can?
No, here, we have your slides. So Darren, why don't you move forward? There we go. We're going to move forward.
Okay,
and then you just let him know.
Can you see them now?
Yep, there we go.
Okay, good. Okay, keep going. And this, this was the uptake in the actual patients. Can I flip it or do you? This was the blood-brain barrier. Next, this was the blood tumor barrier. This was the whole brain. And here's where we are. I'm so sorry. What the lab has found is we have spent 4 years profiling what the blood tumor barrier is. The good news is it's not a chaotic breakdown of the blood-brain barrier. There are consistent differences, 3 different model systems. We see these same differences in human craniotomies. The second good piece of news is we've been able to modulate its permeability just in a model system, but we have changed the permeability of the blood tumor barrier without the blood-brain barrier. We are currently using aspects of the blood tumor barrier to try to deliver drugs. So we have experiments underway that are trying to get more methotrexate into brain metastases using one of these blood tumor barrier systems. And I hope we'll see. This is my blue sky approach. Next slide. Okay, moving to a second project that's ongoing. And this is a lead for the prevention of brain metastases. And those patients who have metastatic disease say, I don't care about that. But I would like to tell you, I think you should. Because as I told you, if a patient gets a single brain metastasis, they're going to get more. It is just time. And so the question is, if you have your first set of brain metastases, can we stop it and not get any more? And that's called secondary prevention. It's a lousy word, but there it is. So next slide. This is the way we have been doing drug studies for forever. We inject the mice, and that bottom green line is after 3 days later, let them recover. We start dosing in drug, And we keep it going. And at the end point, we ask, did they get brain metastases? That is a prevention assay. If that drug stopped brain mets from forming, it's prevention. It is not what we do in clinical trials. What we do in clinical trials is this, and that is you let the brain mets grow, then start the drug and ask, will it shrink or eliminate them? We have now tried 30, maybe 40 compounds. We have 4 with significant preventive activity. We have almost none that have treatment activity. And the drug, to everyone's befuddlement, that worked the best was temozolomide. Next. Temozolomide is a drug that is A, brain permeable, and B, the primary line of treatment for primary brain tumors, glioblastoma, this stuff. So when we dosed it into mice, what you see here are 4 experiments. And experiment 1, can you see my pointer? Maybe? Okay.
No, you can't. So you just have to describe it.
A clinically relevant dose of temozolomide. In other words, it's something that a patient can achieve. What we found is that no brain mets formed. We went down to 25 in experiment 2, no brain mets. We went down to 5 in experiment 3, no brain mets. And we're down 100-fold before we lost significance. Click, please. And then we did the experiment where we gave the mice either a short course of either temozolomide or vehicle. Temozolomide was early or late. And here you have the survival. The vehicle treated mice died on time. And you see that both resulted in long-term cures. Let them run around the cage for months on end. And depending early better than late, you had cures. Next slide. There was a lot of additional information. We didn't do 2 mouse experiments and say, let's do a trial. And I won't go through all that for you unless somebody wants to discuss it. And we are aware that there are previous clinical trials. It says temozolomide doesn't shrink and establish brain met in all comers trials. This would be melanoma, lung cancer, breast cancer. It may have efficacy in combination with radiation therapy, and it has 0 systemic activity in breast cancer. Next slide. And despite that, the mice were saying, hey, we're doing pretty well. And Dr. Alex Zimmer, whose picture I had up earlier, has set up this trial long-term, is the phase 2 trial. It's for HER2 positive breast cancer patients who have already had 1 to 10 brain metastases. In other words, it's secondary prevention. Treat it usually with radiation just to the metastasis. We need a systemic agent, that's TDM1. And so it's TDM1 alone or TDM1 with what we call metronomic, that means every day, low dose temozolomide. Next slide. The primary endpoint is not shrinking anything. It's the percentage of patients that do not get another brain met for a year. This is the phase 1. We're in the expansion phase of dose level 3. We have 1 more patient to enroll, and then I will dance across the stage and tell you the data. Next slide. And I'm not going to go into these. I'm terribly over at this point. But we have put a lot of thought into our exploratory endpoints. We are taking CSF and serum. I want to know, like someone previously said, will CSF predict better than what's going on in serum? We're looking at any number of things. Next slide. Okay, the last thing I want to tell you about is sort of in between blue sky and the clinic. And this is the work of Alex Wu, who is one of the most talented postdocs I've ever had. And it addresses a point that you don't hear about much in breast cancer, although Carrie Anders did some of the foundational work on this. And that is, why do patients who are diagnosed with breast cancer at a young age have a poor prognosis? There have been a number of tumor studies that say, yep, poor prognosis, but they have large tumors or triple negative tumors, or they have an African American race, or they have this or they have that. Maybe that's it. But if you do something called multivariate analysis and take every one of those variables out, At the bottom, at the end of the day, a young age diagnosis is an independent predictor of poor outcome. So we went to the tumor databases and went through them with for those mutational events, et cetera, that were present in primary tumors from young patients. And we didn't find much. Few GATA3 mutations. So if it's not the tumor, is it the host? Which was a crazy idea. It has just about killed us. But what we examined in the host is metastasis. And the way we did it, next slide, is to do young and old mice side by side. We took the same tumor cell model system, put it in young mice, old mice. You'd think no difference. Here is the brain metastasis data. You see 4 different model systems. Young, the number of brain metastases in the red bar, old in the blue bar. Every single time, 3 to 4-fold diminution in the older mice. Click. In the 99LN mouse, they also got liver mets. Don't know why. Don't see that in any of the others. No difference in the liver mets. So young women, young mice get a lot of brain mets. There are clinical papers that say young human breast cancer patients get more brain mets. Next slide. Here is a slide where we looked at 4 different model systems of lung metastasis, and I will spare you the trouble. It's the far right-hand column. There's no difference between young and old. So this seems to be a brain metastasis thing. Next slide. I'm going to summarize a whole lot of data for you. When you know there's something going on in a young brain, we ruled out hormonal effects. In our model system, not saying they don't have profound effects. We then went after the immune system, and we found that there are T lymphocytes, neutrophils, granulocytes, microglia in the metastatic mouse brain. I'm not going to bother you with all the data. What we did is we selectively depleted every single one of them, and nothing with the microglia had an effect on the metastatic rate. And if we, in that A box, if we gave mice a CSF1R inhibitor, which will actually kill microglia, it inhibits monocytes and macrophages, it kills microglia, and then injected the model system, it decreased from red to pink in the lower panel, the number of brain metastases. Well, it's interesting because in normal aging, the microglia, as I said, are the native immune cells of the brain. In normal aging, the brain loses microglia, and with it, they gain activated astrocytes. We see the exact same thing in these mice that we've treated with the Plexicon compound, the CSF1R inhibitor. We see reduced microglia, we see activated astrocytes, and reduced brain metastases. So in essence, what I think we're doing, believe me, we've looked at 1 million markers of microglial activation, nothing's changed. We're basically, I think, making these young brains resemble an older brain, which may not sound like the best thing, but if you're facing a life-threatening challenge, it might be a good idea. This CSF1R inhibitor attenuated metastasis, caused the uninvolved brain of the young animal to resemble that of an older animal. And we wonder, could there ever be age-appropriate therapies, or is one size fits all the way we go? We are working with a pharmaceutical company on what might be an improved version of this. And then we're going to think about how and if to take this to the clinic. Next slide. These are all my collaborators. Thank you so much. I'm so sorry for the foul up. I have no idea what happened. Thank you.
That was great, Pat. And we're okay on timing. And I think it's great that, you're the work that you've done with, the aged mice and showing that correlation with patients, because we see the same. I mean, we all see those observations of the younger, women who do worse, and it's great, that you're leading the field again on figuring that aspect out.
It's killing us. There is, I know people online will know this, there is a Young Survivor Coalition. And I follow their thoughts as carefully as I can. And they're the ones who actually inspired me to go do this.
Yeah, no, we have a lot of inspiration stories. Okay, so while Carrie, and we have questions, but we're gonna hold it off until the end. Thanks again, Pat. Okay, so while Carrie gets her slides up, I'm gonna introduce her. So next we have Dr. Carrie Anders, who's a professor of medicine and the medical director of the Brain and Spine Metastasis Center at Duke University. Dr. Anders received her medical degree at the Brody School of Medicine at East Carolina and did her internal residency and oncology residency at Duke, where she is right now. So she is a true North Carolinian. And so Dr. Anders' ultimate research goal is to uncover the genetic and genomic understandings of breast cancer brain metastasis. And she's been a recipient of many awards to further her research from the NCI, the American Society for Cancer, Clinical Oncology, and Duke University. So Carrie, the floor is all yours.
Thank you so much, Josh, for this opportunity to speak. This has been a great lecture series to date. And it's just so exciting to see what is happening on the preclinical side of things. The insights are fantastic. And as a medical oncologist, I could not agree more. We are seeing our patients younger and younger with brain metastasis, and we need to figure this out for them. So I'm going to take a bit of a pivot and move from the bench to the bedside. The topic that I am going to be covering is the current and future trials for breast cancer brain metastasis. Just a brief disclosure slide. So just to kind of overview what we'll talk about over the next 20 minutes, I think this team has already done a fantastic job of defining breast cancer brain metastasis. I will provide a little historical context of where things were when I started my career from a clinical trials perspective, and then also where we're going in the future. I also just wanted to provide some updates on our current treatment options, because there's been so much movement in this field, both from the local therapy or radiation surgical side of things, and in the systemic therapy side of things. And then really kind of leave with some questions about what we believe the next generation of clinical trials should look like for our patients with breast cancer and brain metastasis. So as many of the prior speakers have said, brain metastasis are becoming a more common problem in advanced cancer. And I know the topic today is breast cancer, but we are seeing this across all of our solid tumor types. We believe that this is because we're doing better at treating our patients' extracranial disease. They're living longer, and we're seeing brain metastasis in diseases we never saw before. I think it remains that our top 3 histologies or cancer types that are associated with brain metastasis are lung cancer, melanoma, and of course, breast cancer. And we do see in our breast cancer population that patients who have HER2 positive disease and those who lack expression, whose tumors lack expression of estrogen, progesterone, and HER2 have the highest risk of brain metastasis during their metastatic journey. So what do we see in the clinic? How do our patients present? We don't have presently guidelines to routinely perform brain MRIs for every patient with breast cancer who walks through the door. And if I had a tool that would help me predict who would develop brain metastasis and ways that I could act on that, I would be so thrilled. So I think that's something we need to be working toward as a research community. When we do have patients, particularly patients with advanced disease, coming to our clinics and we're talking with them about their symptoms since our prior visit, we really try to focus even on subtle changes. word finding challenges. I've had a young patient of mine present with brain metastasis on an MRI just based on changes in her handwriting. Common things that we see based on the brain as the area of metastasis include headache, nausea, changes with muscular strength, and then in extreme cases, we do see seizures and the consequences of increased intracranial pressure. So it's important to think about this in our clinics for all medical oncologists across the disease spectrum, and particularly in the breast cancer space for those who have advanced triple negative disease and HER2 positive disease. And I'm hopeful that we will see the time where we have genomic predictors to help us know when we should be doing routine brain MRIs, because I do believe learning about this earlier in a patient's disease course will improve their outcome. And even better, to echo Dr. Stieg, how could we actually prevent, both from a secondary prevention, but particularly from a primary prevention perspective. So here is our roadmap for local therapy and brain metastasis and decades of research on how we should best deliver radiation therapy to the brain. When I think about this, I think about patients who are presenting with a solitary brain lesion or one brain metastasis, a limited number. And I would say this number starts at 2 and ends at 10 plus. And we're seeing that number rise every year in terms of which patients can receive radiosurgery. And then all the way on the right-hand side of the slide are patients who present with multiple lesions. So we approach each of these different clinical scenarios differently. And we do have data that if a patient presents with a solitary lesion, there is survival advantage for resecting that lesion and then following resection with radiosurgery. If resection is not feasible, i.e. the location could cause side effects that would be intolerable or would leave the patient with neurologic compromise, we do move forward with radiosurgery to that area. And that's not surgery in terms of taking the patient to the operating room, but in fact using very high doses of radiation in a very defined location. And in the center, if a patient presents with 2 or greater brain metastasis, we rely on radiosurgery. And the advantage of this approach is that we can target the lesions themselves in hopes of sparing normal brain. But as Dr. Stieg pointed out, this also leads to brain that's not protected by radiation. And this is where systemic therapies can come in a smart manner to prevent additional lesions in the brain. And then for patients who present with multiple lesions or multiple small lesions throughout the brain, we rely on whole brain radiation therapy, which is typically 10 days of radiation therapy that bathes the entire brain with a low dose of radiation. Because our patients are living longer, which is wonderful, we don't want them to have the longer term sequela of whole brain radiation therapy, including trouble with neurocognition. And so there are techniques to spare our memory center, which is the hippocampus, and to prescribe protective medications such as memantine during whole brain radiation therapy to improve neurocognitive outcomes. So moving forward to the clinical trials space, I wanted to just take a historical perspective to the past. And when I say the past, that's not that distant. I would say 10 to 15 years ago. And when I was in my training and fellowship, and one of the reasons that I elected to move into brain metastasis as my area of research is that I was quite frustrated by the fact that many of our young patients were recurring in the brain. And one of the first inclusion criteria on our clinical trial templates was history of brain metastasis. And so that was limiting, novel emerging therapies only to patients without brain metastasis. And I think we've seen a significant improvement in this paradigm over the past decade. And now we're in the present where we're seeing trials, and we'll talk about some of these trials designed specifically for patients with brain metastasis. And this is endorsed by many of our governing bodies, including the NCI and the American Society of Clinical Oncology. So I'm very happy to see this movement forward. So as a practicing medical oncologist, and when I consider what I have in my toolkit for patients with brain metastasis, we typically go to the NCCN, which is the National Comprehensive Cancer Network, which really combines all of the late-breaking data into a distilled version for practicing medical oncologists. And when we're approaching systemic therapy options for patients with brain metastasis, we largely think about them in two main buckets, including those that are HER2 positive versus those that are HER2 negative, here listed as non-specific. And we'll talk about many of these therapies, but largely in the HER2 positive space, we have a plethora of tyrosine kinase inhibitors. So basically blocking the machinery inside the cells, targeting HER2, in combination with chemotherapies that we know cross the blood-brain barrier, namely capecitabine, and then in some cases in combination with trastuzumab, which is an antibody against HER2, which historically has not passed the blood-brain barrier as effectively. I will say the newest therapy, and we'll go through this data in more detail, is tucatinib, and this was added to the list in the spring of this year in 2020. In the HER2 negative space, you'll largely see chemotherapy here, and this is a space that we really need to be doing better in, and an area of active research of our teams and many others. We have the platinums, and then we'll also talk about the CDK4/6 inhibitors for patients whose brain metastasis are estrogen receptor positive and HER2 negative. So moving forward in the HER2 positive space, this is really just another view of the therapies that we've just discussed, but the ones that I really want to focus in on today are the newer kids on the block. So the tucatinib compound, which was defined through the HER2CLIMB study that was mentioned earlier, as well as a novel antibody drug conjugate called trastuzumab deruxtecan. So tucatinib, as I spoke of, is a small molecule inhibitor that targets the tyrosine kinase intracellular machinery of a HER2 positive cell. And one of the uniquities of tucatinib compared to the earlier generation tyrosine kinase inhibitors is that it is very specific for HER2 and does not hit another target called HER1 or EGFR, epithelial growth factor receptor. And what this does is it really limits the toxicity to patients, where we do see GI toxicity and rash in particular when we target EGFR with the neratinib and lapatinib compound. In the laboratory, HER2 positive breast cancer brain metastasis mouse models were evaluated to each of these three therapies, and you can see in the, trying to see the color here, oops. In the tucatinib arm, also, or the green, previously known as ONT-380, the best survival of that animal model, which then led into the phase 1 experience of capecitabine. We talked about this being a brain permeable chemotherapeutic with the HER2-directed antibody trastuzumab and the tyrosine kinase inhibitor tucatinib. And what was interesting in the early phase experience is that we saw beautiful responses, 61% in the extracranial disease space. But what was also quite interesting, knowing that this drug crossed the blood-brain barrier, the central nervous system or brain metastasis response rate was also quite respectable at 42%. And just to give some historical perspective, in the past, you know, kind of our gold standard of lapatinib with capecitabine, response rates were around half at about 20%. So this moved forward to the design of a phase 3 randomized placebo-controlled study called HER2CLIMB. And this was a study of over 600 patients, all with metastatic HER2 positive breast cancer. And one of the very unique aspects of this study, sort of thinking about where we were 10 to 15 years ago, where patients with brain metastasis were not enrolled or were excluded from these studies, patients with brain metastasis were enrolled in this study and actually made up close to 50% of the patient population. The patients were randomized in a 2-to-1 fashion to tucatinib trastuzumab capecitabine versus placebo trastuzumab capecitabine. And here are the results that we learned about last December. We did learn that the HER2CLIMB primary analysis, which included all the patients, those with and without brain metastasis, not only had an improvement in progression-free survival, or the time before a patient's disease became refractory to that therapy, that risk of progression was reduced by almost half. The other thing that we learned, and importantly, is that the risk of death was reduced by a third for the addition of tucatinib to the capecitabine and trastuzumab. The patients on the tucatinib arm were living longer. And what I don't show in this slide deck, but was the toxicity profile. And while we did see some GI toxicity, it was much less than previous generations of therapy. And then when the group of patients who were enrolled with brain metastasis, so 291 of the 612 patients, there was also a significant reduction in progression of death by 50%. So this was very exciting and led to the FDA approval of the triplet combination, which we're using routinely in our clinic today. So I think this is a beautiful story of really a bench to bedside for a patient population who clearly is in need of approved available therapies. And I'm just thrilled to be able to have this therapy at my fingertips in my clinic for my patients who present with brain metastasis. So moving forward with an antibody drug conjugate. So this is trastuzumab deruxtecan. And essentially what you'll see here on the slide, on the left-hand side of the slide, you'll see an antibody, just like antibodies against viruses, but this one is targeting HER2. And it's linked to chemotherapeutic, which has a mechanism of a topoisomerase inhibitor, so essentially interferes with DNA, it induces DNA damage. And what's interesting about this particular compound is for every molecule of trastuzumab, there are actually 8 moieties of chemotherapy, or 8 different chemotherapies or payloads, so a very potent payload. If we tried to give that degree of chemotherapy directly into the vein, it would not be tolerated. So this is essentially a Trojan horse effect, where the trastuzumab binds the cancer cell and delivers this very toxic payload directly to the tumor cells, sparing the patient the toxicities. So this compound was evaluated in a series of studies and is ongoing as well, called the Destiny studies. And we learned about it initially in the Destiny Breast 01 study. Essentially, this was a study of over 250 patients who had advanced HER2 positive breast cancer, who were treated with several different doses, which was deemed, the final dose was deemed 5.4 milligrams per kilogram. But what was also interesting here is that patients with stable treated brain metastasis were allowed. So we're seeing that theme. So essentially this slide shows that almost every single patient enrolled in this study had reduction in their tumor. So all of the, this is called a waterfall plot, all of the blue lines represent reduction in tumor by a certain percentage. And for those who had reduction over 30%, what we would call a confirmed overall response, this was 60%. And then I took this text directly from the New England Journal of Medicine paper to just show the magnitude and the duration of response to this therapy in patients with brain metastasis. So essentially what I want you to take home from this is that the patients who had brain metastasis at enrollment did actually even a little better than the patients who did not have brain metastasis. They were free of disease progression for 18 months, a year and a half, before having to move to another therapy. The other thing to note about this patient population is they'd received 6 prior lines of therapy, so a really unprecedented progression-free survival. So very excited about this compound. So I want to be cognizant of time, but also wanted to just briefly talk about systemic therapy for HER2 negative breast cancer brain metastasis. And as I said earlier, this is a space where I'm hopeful to see more development in the coming years. Whereas we do have FDA approval of tucatinib in patients with brain metastasis and the trastuzumab deruxtecan across metastatic breast cancer, we don't have any FDA approved therapy specifically for patients with HER2 negative breast cancer brain metastasis to date. But I did want to focus in on one compound called abemaciclib. This is essentially a small molecule that blocks cell cycling, which is seen in our cancer cells, And it is specifically approved for patients who have estrogen receptor positive, HER2 negative metastatic breast cancer. One of the things that we learned about preclinically is that this compound crosses the blood-brain barrier. And we were lucky enough to be part of a study called JPBO, which investigated the activity of abemaciclib in patients who had HER2 negative, but hormone receptor positive metastatic breast cancer to brain. And we did see that many patients benefited from that therapy, and we use this routinely in our clinics. And when I am selecting a CDK4/6 inhibitor from the group that we have of 3 in my patients with brain metastasis, this is my first choice. So Dr. Stieg also talked about Priscilla Bastianis's work at Mass General, and she shed a significant light into the biology of brain metastasis across all histologies. And this is just an example of one of her set of data showing alterations in the CDK pathway, the target of abemaciclib, in over 50% of the brain metastasis, which could potentially predict sensitivity to a CDK4/6 inhibitor. And in fact, this has been translated to the design of a clinical trial that is enrolling nationwide. So this is essentially a match type study where patients who've had a craniotomy or a brain surgery for standard of care purposes could have their brain metastasis tissue tested for alterations in the CDK pathway, the Pi3 kinase pathway, or the NTRK-ROS1 pathway. And if that alteration is found in the brain metastasis, they would be assigned to the appropriate treatment arm with brain permeable compounds that target the alteration. So as we look toward the future, and I know I've given a lot of information here, there's still many remaining questions. I did not touch on immunotherapy in my talk, others have, but I think one of the questions is how will immunotherapy impact our patients with brain metastasis? Many of our solid tumor types have an immunotherapy indication. So now we need to see how are both the extracranial disease compartments and the intracranial disease compartments responding, and how might it differ in the brain microenvironment, just as we've heard of earlier today. There's a lot of emphasis on who should go first. Should it be the radiation oncologist or should it be the medical oncologist? And are there benefits to sequencing radiation therapy before immunotherapy or vice versa? We're also looking at combination therapies for brain metastasis. So in the setting of HER2 positive disease, should we be combining tucatinib with other therapies in the HER2 positive space? And in fact, there's an ongoing study of an antibody drug conjugate called TDM1 with or without tucatinib. And then also the concept of a window of opportunity study. So that is evaluating a therapy or a systemic therapy, drug therapy prior to a resection of a tumor for standard of care purposes to see how much drug is actually getting into the brain metastasis to complement much of the work that we've seen in our animal models and many, many more. So with that, I'll close. I really appreciate the opportunity to speak to you today. There is so much to discuss in this space, and I'm very happy to see where we are, a decade plus later, and only am even more excited to see where we'll be a decade down the road with all of the exciting preclinical data we've seen for translation. So thank you, Dr. Niemann, for the opportunity to speak. Look forward to Q&A.
Thank you, Dr. Anders. It was great to hear all the progress. And I know that this is a big concern for patients, about wanting more clinical trials and brain mets, and that certainly has changed over the landscape of years. And I know that you have been a huge advocate of this. So I know everyone has been, very appreciative of the push that you've been doing. Okay, our last speaker of the day, but certainly not the least, and is I'm actually looking really forward to hearing her speak, is Dr. Kathy Van Dijk from UCLA. Hi, Kathy. Carrie, can you stop your slides and then sharing, and then we can get Kathleen up? There we go. And while she puts up her slides, I will introduce her formally. So Dr. Kathleen Van Dijk is an assistant clinical professor in the Department of Psychiatry and Behavioral Biosciences at UCLA. she got her PhD in psychology and neuropsychology from the City University of New York, and then did her externship at Columbia University Medical Center and a subsequent postdoctoral in geropsychology at the Johnson Cancer Center at UCLA. Her area of expertise is cognitive function in breast cancer survivors, which is a huge issue, and I'm looking forward to as a neuroscientist myself. And she's been a recipient of grants from the NIH and the American Cancer Society, and really looking forward to your talk. So the floor is all yours.
Thank you. Thank you very much. We're really excited to be here. And it's an honor to speak with such distinguished panelists. I really enjoyed the talk so far. So I am going to be giving an overview of what we know about cancer-related cognitive problems. And largely this literature is in the area of non-metastatic disease, and a lot of what I'll be presenting is from breast cancer survivor studies. That being said, I think it's an area, metastatic disease is certainly an area that needs more focus and needs more attention and better understanding how the metastases and treatments can interact with the brain and change functioning. And I appreciate Dr. Steegs and Dr. Anders' comments in that regard. Okay, so I'll get to it. So I'd like to start out discussing cancer-related cognitive issues by thinking about the history. And this is what I find fascinating, is that this area has really been driven by patient report. And so what you're seeing here on the right is 5-year survival rates, which have been going up largely across most types of cancers. And you'll see this kind of dramatic increase in the number of articles that you see on PubMed and online about cancer-related cognitive impairment. And so a lot of this work began with the term chemo brain, and I'll talk a little bit about why that's a bit of a misnomer, why we're switching over to cancer-related cognitive impairment in a minute. But I think it's just important to recognize the issues have been exponentially getting attention. Yep, hang on. There we go. Okay, so, there are a wide range of reported rates for cancer-related cognitive problems, and just giving you kind of a smattering here from the literature. So within the past year from treatment, the sample that was surveyed for the Livestrong study, 45% reported that they had cognitive problems in the last year. In the sister study of breast cancer survivors, 60% had reported that they had some kind of cognitive issue after treatment. And what I find particularly disheartening about that study is that of that 60%, only 37% had actually talked to their provider about it. And so the authors had gone on to then suggest that maybe there was some discomfort, or they didn't feel like they wanted to bother, quote unquote, their doctor with these problems. So I think there's a real need to elevate the awareness about these issues and discussion. The field in general has landed on a number of about 20 to 25% of non-CNS cancer survivors who have some kind of cognitive difficulty or some kind of lasting cognitive problem after treatment. But it's still we still have a lot of work to do in terms of characterizing it and really getting down into what's going on. The reported cognitive problems and the cognitive issues that have been documented with neuropsychological and neurocognitive testing have been backed up by neuroimaging evidence. So I'm just giving you a bit of a survey here of some of the neuroimaging studies. So in panel A, all of these are looking at before and after cancer treatment or chemotherapy, excuse me. In panel A, you're seeing changes in gray matter volume, so a shrinkage of the gray matter in the brain. In panel B, you're looking at white matter tracts, the highways of the brain, and there has been some degradation of the integrity of the white matter tracts after chemotherapy. And in panel C, looking at functional activity, doing a working memory task, and changes in how much energy the brain needs in order to achieve that task. So we're seeing a signal and we're hearing from patients that they're having problems, and we're seeing changes on testing. So the field as a whole is really working to wrap its arms around what's going on. Just to give kind of a sense of the types of issues that patients describe, and this is often what I hear clinically when I see patients, that there's this fine degree of memory or speed of thinking that changes. They struggle to remember things. They feel not as sharp, that their edge is gone. They feel mentally spent. A lot of times, patients will report that they are having problems coming up with the right word, or that they can't remember a name as they used to. So it's typically not a dramatic change in cognitive functioning, but that doesn't mean that it's not one that can be very distressing and disruptive. It's typically more mild and subtle, but it is certainly something that can be that can interfere with functioning and quality of life. So breast cancer survivors, who have these problems, talk about concerns about their job performance. They have reduced self-confidence. They feel overwhelmed. And what the literature has said is that it would have been nice if the patients had been informed that this was a possibility before treatment, so they could plan. And I think that that's a really important comment from a patient, because the surprise of having these difficulties after treatment is over can be incredibly distressing and confusing for patients. So initially, when researchers started looking into the question of what was going on in non-CNS cancers, the toxicity of chemotherapy was thought to be the main culprit. And it certainly is still one of the primary culprits. So there's some studies that have shown, changes in cognitive functioning 20 years after exposure to chemotherapy. But as the chemotherapy regimens have gotten less toxic, less neurotoxic, we started to see a bit of a reduction in the severity of those symptoms. That being said, the researchers are also looking at various other aspects of cancer treatment and how those can impact cognitive functioning as well. So there are studies that report changes in cognitive functioning after radiation. Some studies have reported changes in cognitive functioning even before treatment. And one of the things that I've been particularly interested in what I've been developing my research lab in, is understanding the effects of endocrine therapy on cognitive functioning. So I'm gonna take a little bit of a detour and walk through that for a minute. So as I'm sure most of the audience knows, about 75% of breast cancer patients have hormone receptor positive tumors. So the estrogen is feeding the tumor. And these are then treated with aromatase inhibitors, which reduce the endogenous estrogen in the body, or tamoxifen, a selective estrogen receptor modulator, which blocks an estrogen receptor, but doesn't change the amount of estrogen in the body. So one of the studies that I worked on with my mentor, Dr. Patricia Gans, was looking at neuropsychological functioning in women based on whether or not they received endocrine therapy after their primary treatments, meaning chemotherapy, radiation, or surgery. It was a prospective naturalistic study, so there's a lot of error in those types of studies, but really we were just following these women from before they may have started endocrine therapy, 6 months after, then 12 months, and then there was a follow-up about 3 to 7 years later, depending on when they were enrolled in the study. So we really got a good sense, a good swath of time to look at neuropsychological functioning in these women. And what we found was nothing. So we found that there were basically no differences in neuropsychological functioning based on whether or not someone had been exposed to endocrine therapy treatment or not. And so I looked at this data every which way, because I know that this is a complicated question, and prior literature has shown some changes. Some other researchers have found no changes. So I think there's still a question about what's going on. So we, thinking about the results of this study and knowing that there is significant report of cognitive changes for some women on endocrine therapy, is am working on developing a study right now that's looking at different aspects of what hasn't been looked at before. So neuroimaging, maybe the measurement sensitivity isn't good enough. We're not quite sure what the mechanisms are. We know that these agents cross the blood-brain barrier. We know that there are estrogen receptors all over the brain in important areas. And no one yet has looked at the impact of the woman's menopausal status on as a predictor of whether or not they will ultimately have any cognitive problems as a result of these treatments. So bringing it back to the broad conceptualization of cancer-related cognitive decline, the field right now is really looking at this problem as a multifactorial issue, that there are a lot of different factors that can go into whether or not someone is experiencing cognitive changes. So of course, including cancer and cancer treatments, but also other comorbid conditions that the patient might be dealing with, like cerebrovascular disease. Stress and depression is a big part of it as well, in terms of, we know that stress can have a negative impact on cognition, both in terms of the hormonal effects, but also just the neurocognitive aspects of the internal thinking and internal depression that can happen. And I talk about that a little bit later. Sleep disturbance is a big factor as well, something that we're looking at more closely. Cognitive reserve, the idea that someone brings with them a certain amount of resilience against these issues. So it's an evolving tale of understanding what's going on, but this is a kind of an overview of why we think these issues might occur. So one of the ongoing models is the idea of accelerated aging, the idea that these cancer treatments are actually, through a variety of mechanisms, inducing a bit of an acceleration in brain aging, because we see similar types of aging-related patterns in the types of symptoms that someone develops. And so it's not proven, quote unquote, yet. It's still the ongoing model, but it's consistent with the idea, that was discussed earlier in terms of neuroinflammation and oxidative stress and a variety of overlapping mechanisms in brain aging with what we see in preclinical and clinical models of cancer-related cognitive decline. So that this model is also kind of explaining why we're seeing these overlaps, what we're seeing in terms of overlaps. So the idea that there's a acceleration of the reduction in telomere length, which is the genetic cap that then allows a cell to reproduce, increased oxidative stress, DNA damage, which we found in our study as well, was associated with cognitive problems and cellular senescence, cellular death. And so there's similar overlaps in these two issues. So we're using aging as a model to try to understand what's happening for patients who are experiencing cognitive problems. Okay, so another area of research that I've been working in and I'm very interested in is trying to understand what we can do about it, both in terms of prevention and also treatment. The bottom line is that more research is needed. There's a handful of studies out there right now that have looked at this, and so there's ongoing development. But what I'm going to do is just walk through a couple of the known ideas or the more promising ideas that are out there in the literature right now, and then talk a little bit about some very basic strategies that I might review with a patient in terms of cognitive rehabilitation. So there are guidelines for asking and approaching patients and assessing for cognitive difficulties. These are the ASCO guidelines, referring patients when they have problems, and assessing for reversible contributing factors, which we'll talk about in a second. So there are guidelines, and I think that the stats that I showed you earlier are kind of getting at the idea that this isn't something that's as widely discussed and brought up as perhaps could be. So there's a lot more work to be done in that area. In terms of treating contributing factors, so things, we think about, well, what what can we do something about in that kind of multifactorial model? We can't take away cancer and cancer treatment. That's done. That's history. So, but perhaps we can elevate and improve some of these other contributing factors, and that will lead to an overall improvement in cognitive functioning and well-being. So things like improving sleep. There's a study that found good efficacy for an app, a cognitive behavioral therapy for insomnia app in breast cancer survivors. So de-stressing can also be very, very overlap with sleep as well. So things like meditation, mindfulness, exercises, yoga, and increasing or sustaining physical exercise. So, it's not news that physical exercise is good for you. And there are a few studies that are going on right now, and a couple of small studies that have found some improvement in cognitive functioning after the introduction of more physical exercise. The other possibility for treatment is cognitive rehabilitation. And there are a handful of studies that have looked at cognitive rehabilitation in cancer survivorship, and they've all found pretty good results. So for those who may not be aware, cognitive rehabilitation involves a lot of different components: education, reframing what's going on, meaning trying to change perspective so that we can approach a problem in a different way. Distress management is a big part of it. So the distress that can come from experiencing a perceived cognitive problem, and compensatory strategies. So I used to be able to just go to the store and buy, a gallon of milk without needing a list, but now I need a list. At the end of the day, I still get milk, but I had to use this compensatory strategy. Okay, so I wanted to give you just a flavor of what some of these strategies look like, and what I do when I talk to patients. So really, focusing in on the individual patient's needs. What does their desk look like when they're working? Do they have dings all the time from emails? Really trying to help patients clean off their physical desk and their mental desk to help them focus and improve attention. And making sure that they have carved out time, that they're not to be disrupted, having a do not disturb sign, communicating with other people so that they're left alone and they're not distracted, organizing their time so that they have particular chunks of time to address certain tasks so that they don't feel like they have to do everything at once, because multitasking is often something that patients struggle with. Helping to use basic memory aids. So how do you maximize the use of your phone or the use of your calendar, or just a plain old paper and pencil, in order to get where you want to go, in order to function the way that you want to function. And lastly, one of the components that I've been working on researching and also incorporating into rehabilitation services is the idea that I've seen clinically much more often than not, the idea of this loss of self-confidence that accompanies experiencing cognitive changes. A professor giving a lecture and not being able to come up with a famous researcher's name off the top of the tip of their tongue. So it really can be a very unsettling issue for a lot of these patients. And so we'll work on positive self-talk, really generating just some internal monologue of that it's okay in order to move on. And so the research is looking at, some researchers have found that there's a trauma aspect to the development of cognitive difficulties. And so we'll work, this type of intervention helps to work on that as well. So there's a lot of different ways that we look at to help patients. And like I said, we're still in need of a lot more studies and a lot more data, but we're trying to come to an understanding of why these symptoms occur, and then how we can help patients function and have optimal quality of life if they are experiencing these symptoms. Just some more references, and I'd like to thank my primary mentor, Dr. Gans, who's been wonderful, and as well as the patients who volunteered their time for the research that I've worked on, and my funding institutions. Thank you all for your attention.
Thank you so much. I mean, I think this is great to have you on, you know, on the panel as we're going to proceed to it. I think it's a lot of patients, you know, have these cognitive issues, and I think this is going to be a emerging field, you know, even more in the years to come. Okay, so we're only a couple minutes over. I'm thankful. So now we're going to go through, and I have some list of questions that we've collected from, you know, all of the feeds that we have, and we'll have. I'll ask the panelists. So one of the first questions, and we'll go in the order, I guess, of the talks, is for Dr. Jar Sears. So someone asked, does dormancy occur everywhere? And if it does, are all the cells the same type?
I mean, it's a wonderful question. It's really currently a very difficult one to answer because answering that question would require really a wholesale sampling of the body. And that's something that, you know, has not, you could attempt at autopsy, but, you know, we haven't taken that, that extensive of a step yet. If I had to guess based on, you know, putting together a bunch of data and integrating studies that have been done in mouse, I would guess that there are disseminated cells throughout the body, even in tissues where we never see metastasis. And I would also guess that there are differences between the states that these cells are in. I think some could be senescent, some could be, you know, quiescent, meaning that they actually could be woken up. And I think that there's a time component that the longer these cells are in this state, the harder it is for them to wake up as well.
Great, thank you. Dr. Priceman, so one of the questions that came through, I know it was two questions for you, but one of the questions that came through was, does HER2 positive CAR T cells, can it be used to prevent brain mets if a patient doesn't have it yet?
Yeah, that's such a good question. You know, we started the trial with a thought that we would treat patients with active brain metastasis. We're now in the process, based on the safety and what we've learned in terms of the activity of the therapy, in opening a new trial for patients without brain metastasis, but with systemic metastasis, in treating them with intravenous delivered HER2 CAR T cells. And the idea would be that these might be high-risk patients for brain mets and or leptomeningeal disease, and we'll learn a lot from that. But the ultimate goal will be to start treating patients with and without brain metastasis by different routes of administration. It's a tough concept, and it's really tough to show that clinically with the number of patients that any one trial in the early phases enrolls. But what a good question. I mean, we hope that we could eventually prevent dissemination of disease.
Yeah, and the other question that came up in the chat box, you know, is someone asked, what's the difference between intrathecal delivery and intraventricular? And I know, I mean, I'll let you go. I mean, we can, all of the panelists can chime in, but can you tell us a little bit about your experiences with your studies and the differences in those, if you've done them?
Right, so we haven't tested that specifically. Intrathecal and intraventricular enter the same fluid, but there are differences, and people have observed that in the context of other therapies. I mean, it's beautiful. Actually, Pat can probably answer that better with tracking different therapies in her studies preclinically, but we haven't tested that. It's something we thought about in treating leptomeningeal disease more effectively. Our radiation oncologist at City of Hope has suggested that on a number of occasions in treating patients with lepto or brain disease that are in particular locations. And so we are aware that there may be differences, even though they enter the same base.
Yeah, great. Dr. Stieg, the next couple of questions is for you. So someone asked, does menopause do more to fight age-related metastasis than just lowering selected hormones?
Well, that's going to be particularly difficult for me to answer because mice don't undergo menopause. It's, you know, they're a little different from us. Now, what Diana Satelli in Colorado has done is to try to artificially put these
Gotcha.
mice in menopause with a variectomy. Whether that mimics to the best degree or not, I don't know. She's been doing that and then supplementing with this hormone or this drug or that drug. And so we might get at the answer, but unfortunately I can't answer it with any data that I've generated.
Yeah, no, that's a great question. The other question that came up, which is, do younger tumor cells go more to other sites, or is it just to the brain? And I know you showed that.
So in our hands, now, I can't tell you how old these tumor cells are because they're cell lines that have been in culture. So it's a younger host or an older host. We did liver, lung, and brain. And of those three, it was only brain. I can't tell you that it won't be other organs as well.
Got it. Okay, great. Dr. Anders, so we have a couple of questions for you. So one of the questions in the chat box is, lobular cancers can go to the leptomeninges. Can you talk about treatments for this, and should metastatic lobular patients get routine MRIs?
That's a great question. So about 30% of breast cancers are lobular. About 60% are ductal and arise from the ducts of the breast. And there's about 10% of other, which are more rare types of breast cancer. One of the hallmarks of lobular breast cancer is there's a lack of an adhesion molecule between the cells called ecadherin. And in fact, that's what our pathologists look at to determine if a tumor is lobular or ductal. So by nature, instead of grouping together in more of a spherical metastasis, lobular breast cancer tends to line organs such as the pleura, the coating around the lung, the peritoneum, the coating around the GI tract, and then the leptomeninges that protects our brain. So I think we certainly see this in the clinic, and I think another thing that we really need to understand is how should we be treating lobular breast cancers differently than ductal? And I will say right now in the clinic, they're treated relatively similarly, but based on their biology, they probably, I think over time we need to develop better strategies. In terms of treatment of leptomeningeal disease more globally, I think essentially there can be kind of two types of leptomeningeal disease. There can be diffuse carcinomatosis, where we see this sort of, we call it a sugar coating of the leptomeninges, which is just a fine film of tumor cells lining the leptomeninges around the brain, and that can also go down the spinal cord. We're also seeing nodular leptomeningeal disease, which has a different behavior. So more discrete areas along the dura. So I think the first thing there is diagnosis. We use MRI, brain MRI, and then we also use sampling of the cerebrospinal fluid. And if patients are symptomatic, with either headaches or nausea, dizziness, we give steroids. And our first line of defense is usually whole brain radiation therapy, really just to halt the symptoms, really treating the symptoms. And then moving into the systemic therapy space, there are several different options. You can use systemic therapies that have a better chance of crossing the blood-brain barrier. I saw one of the questions in the chat box was around abemaciclib. In fact, we do have some data for abemaciclib with leptomeningeal disease. We also have, and it's been really interesting to hear the conversation around intrathecal versus intraventricular therapy. Typically if we're going to do what I guess I've previously called intrathecal therapy, we do it through an omaya, which is a port placed on top of the skull, but actually burrows down into the ventricle so that we can give repeated administrations of chemotherapy, sometimes twice a week, as opposed to having to do repeated lumbar punctures, which can cause a lot of challenge. So in essence, we're probably actually giving intraventricular therapy when we give intrathecal therapy. So it's just something that's been interesting to hear today. And I think, I think right now we typically give chemotherapy, methotrexate. We've also seen data for trastuzumab into the CSF, and I'm very fascinated by the CAR T cell delivery into the CSF. So I think there's a lot of development here yet to see, but those are kind of our initial decision trees in the clinic.
Yeah, that's great because I mean, what you described of even within, you know, the location of the brain mets, whether it's, you know, in the front or the back, that actually really, you know, addresses that concept of the tumor heterogeneity, right? These tumor cells have now changed and it gives, you know, it gives some tumor cells proclivity to grow here versus back here. And we've, you know, we've had studies underway looking at patients as well, looking at what we call topography of brain mets. And
The
so,
map of
yeah, the
exactly.
map of the brain, you know, which is very, very important. And as far as, yeah, I mean, lepto, yeah, I mean, intrathecal versus intraventricular, I mean, it's just different hall, you know, it's just different rooms, but they're all connected by the same hallway. So, but it's also, yeah, like what you described is trying to give patients the best route so that they don't get infected while getting therapy and the pain. Can I ask you, and you addressed a couple of other questions within yours, but one overall topic, you know, there's a long question that came up, but, you know, and this by far is, you know, of course, is not medical advice at all, but could you give, you know, as a medical oncologist, you know, if a patient was diagnosed, and this was, I'm abbreviating this question, you know, if a patient comes in, you know, and they find out they have metastasis, could you give 1 or 2 pointers of what should they be asking their doctor next? Of course, it's not medical advice, and every patient, you know, needs to consult, but just if you can give 1 or 2 simple questions they could ask.
Sure. So I think one, understanding the subtype, right? Because so many of our decision trees involve subtype, you know, just the way that I organize my presentation. You know, we think about, you know, breast cancer is not just breast cancer anymore. It's multiple different types of breast cancer under one umbrella. So really understanding the type of breast cancer that an individual has is very empowering and really helps understand the treatment algorithms and the rationale. I think the other thing that has really been a paradigm shift in how I trained in oncology and how I practice now is going one step beyond subtype to next generation sequencing. And when I see my patients in clinic, I always offer them next generation sequencing on preferably a metastatic tumor biopsy. And what that means is essentially getting gene alterations, whether or not it's mutations, deletions, amplifications across hundreds of genes. And that really helps guide our therapies now. You know, we have FDA approvals for therapies that target estrogen receptor positive, HER2 negative, Pi3 kinase mutant breast cancer now. We're picking up alterations in the BRCA genes that the patient didn't inherit, but actually happened in the tumor itself. And that has shown some activity. The PARP inhibitors have shown activity there. So I think, you know, really number one, understanding subtype, but then also going the next step to understand the molecular changes in the tumor. And that's something I would just, if the physicians haven't offered in the metastatic setting, would be something that I would certainly ask about, because many of these tests are FDA approved and covered and have been, you know, they've really changed the way I practice medicine.
Great, thank you. And last question is, you know, Kathleen, you brought up this idea of, you know, this whole, you know, cognitive, you know, decline. If a patient wants to come in, or if a patient wants to, you know, act as a healthcare worker, or if, you know, a doctor like yourself, how, you know, would they be talking to, you know, their oncologist, or how would a patient, if they're not at UCLA, for example, that was one of the questions that came up, you know, ask?
Yeah, so it's a really good question, and there certainly aren't as many providers as I wish there were who are assessing and addressing these issues. So I think, you know, seeking out help from rehabilitation specialists like occupational therapy, speech language, if those issues come up, and neuropsychology. So asking if those resources are available wherever they are. And for an assessment. I think it's important to keep in mind, though, that what we find in the literature is the types of problems that people have often don't meet any criteria for a neurocognitive disorder diagnosis. Doesn't mean that they don't exist and that they're not problematic and need to be treated. It's just a bit of a fine line when, you know, you see someone and their cognitive performance looks okay, it might be that they've had a decline. It's just not so severe as someone who has dementia or something like that. And so I think it's important for patients to keep that in mind, that their concerns are valid no matter what the actual objective testing says.
Right.
And that, I wish all providers had training in cancer, but if they don't, your concerns are valid. And, and just continue to find someone who you can work with, like someone in the rehabilitation community.
Okay, great. Thank you. Okay, with that, I know we're, about 10, 15 minutes over, but I really want to thank all of you, Pat, Carrie, Kathleen, Cyrus, I see Alana there, Saul, who had to go to another meeting. Really want to thank you all for, your wisdom and your pearls, and updating, all of those participants that were on. I hope that, we're making lemonade out of lemons. I hope that, even, we'll be back to see each other at conferences in the future, but I hope that we could still do this. Like Zoom has, this one positive aspect of COVID is now we've all become virtual folks, right? And we can, continue these conversations with patients all around, I'd say the world, because, that's where this is reaching, and patients could benefit, from these kinds of conversations. So I want to thank you all again, and to all the folks that are out there. And I hope you guys all got a very useful day out of this. And hopefully we'll see you soon. And I hope everyone stays healthy. Thanks, everyone.
Thank you, Josh. Thank you all.
Thanks, Josh.
Okay.
Take
Bye,
care,
guys.
everyone.
Bye.
Bye. Bye.
Bye.